So I welcome you all to our second lesson of cell biology, strictly handling section 3, that is cytology, where we are looking at the outer cell structure. And I remember introducing you in the very first class where we talked of prokaryotes and eukaryotes as the major types of cells. So I believe in session 1, prokaryotes were exhausted. So we are here for eukaryotes to handle the biggest percentage of them, leaving some simple concept behind that will have to be taken by you as research work, okay? So you all know that we always have new members that join us in each and every class. And I'm always requested by my team that I talk about our sponsors for the free class. It's really protocol. So those who have really over listened to this will forgive us, but it is the protocol. We are supposed to welcome the new members and we also let them know of the people who sponsor these free classes. The sponsors, it is Science Scholars Academy Uganda. So it has sent me SSA to take you through eukaryotes under cytology and cell biology. Hope that is okay. So as usual, give me a minute or two. I talk about our sponsors, what they do, how to get in touch. Then we begin our class successfully. So thank you for the chance. Okay. Now, very fast, those who are having this as their first class at SSA, I'm Dr. Paul, or you can call me just doctor, just like most students do, and it's very okay. I like it. Yeah. So in case you're having a medical complication or anything, don't hesitate to reach out. I'm just a medical doctor who is passionate about educating young generations. Okay. So like today someone came to me and told me, doctor, I'm sick, I have allergies. I told that person allergies are not a disease. They are just complications for various diseases. So I advise them to visit the physician so that they take some history and they get to know which kind of medication is really perfect for them. So anytime you have any medical complication, please don't hesitate to reach out. I'm a medical doctor, passionate about educating the young masses. Yeah. And my favorite subject is chemistry. Yeah. Be aware. Now, the people who have sent me to really take you through uh, are the owners of Sun Scholars Academy. They have told me, doctor, come and take through these senior fives, eukaryotes, because they over requested for it. Then I said it's okay. So SSA has a vision, a mission, and whatsoever of creating competent science scholars in the country. And they told me to also tell you that they handle only and only 3 services. Outside those services, it is outside their concern. So the very first one is the holiday coaching program that is compulsory to all science students of the country. And they told me that it goes for as much as or as less as 100,000 for all combination plus the subsidiaries, the sub ICT, the GP, the sub math, things like that. Okay, so your whole combination is just handled at that. Then the homeschooling program, and they updated me that this is for those students that don't have fees, those with health issues that stop them from attending school, and those with other issues. I don't know what are those issues. Maybe those people have cultural possessions. Maybe. But they said even those other issues that stop people from schooling, they can really incorporate them into the homeschooling program. Then the other free sessions are those of counseling. Please, the counseling should be purely academic. Okay? Then advice and consultation, just free of charge for everyone in the country. So currently, as of 2026, the video lessons provided are for the 4 major science subjects. That is biology, chemistry, math, and what? And physics. If it is a live class like this very one, it is strictly via Zoom. And when it's recorded, because people miss these classes for very many reasons. They don't have data, they don't have battery, they don't have network. They are just moody things like that. So we record and keep them on our app. But we usually use Google Drive because it is more student friendly, okay? So the other things, we don't sell notes. Most of you have really asked me for notes for some good time. But in the future, I'll tell you where you can find the bookshop of SSA. Then you can get those books from there. I can even give you a coupon. If you're my friend, you can get books for free. Yeah. But as the curriculum has not yet been so much understood as far as assessment is concerned. So by the time the curriculum is perfect, those books will be there for your siblings. Okay. So you can just take a screenshot as usual. You can go through the rest anytime. To get in touch, that is our contact. The subsidiaries, I told you that they are also handled. The recorded classes since 2015. They are refined and perfected, and they are available. So you can access them anytime throughout the year. That is our email in case you want to get in touch as a corporate person. Then also our YouTube channel. You can visit it for the free sample classes. If you really want to sample a few things here and there, it is always there. And don't forget to subscribe. Don't forget to share with your friends. So those are our sponsors, Science Scholars Academy. They are sponsoring the free class. Those who have overheard this, yes, I have nothing to do for you. That is it. Now, very fast, we go to the syllabus book. We see what we've covered, what we are going to cover today, what you're taking as research, what our next stop is. And yeah, now, cytology is the section 3 competence for the cell biology branch. Okay. And you all know we are here to analyze the ultrastructure of whatsoever and do some sort of comparison and distinction. Okay. So the other time we handled prokaryotes very well. So for today, we are here to handle the animal and plant cell. So trust me, I'm not going to leave this class. You ask those of physics. I can even leave at 4 a.m. unless I'm done with these two. So what I really want you to take home as research is this: the fluid mosaic model for the plasma membrane. Yeah, you're taking it as research, and it will also have presentations that I will attend to, give you scores, and then we also have it as a class. But that one may be premium and I may not change my mind. Okay. Then these other things I think we covered, if you can recall. The eubacteria and archaebacteria. We talked of the ultrastructure. We talked of the categories. Even today we are going to categorize the eukaryotic cells, things like that. So make sure that you listen and make sure you have a very keen eyesight, because there are things I'm going to be exposing and explain. Expose, we explain. So the same format we've been using for those classes, it's the same format we are using today. But in the future, we shall have a format that involves a textbook and then other things that I said the other time. Okay, so let me hope we are set. Now, in our workbook, someone wrote in the screen, so please don't misbehave. Yeah, don't misbehave by doing that. Be disciplined. Okay. Hope the screen is back. Now, the lesson targets. The moment I write lesson targets, just have it in mind that I'm not going to leave the class unless they have been dusted. Okay. So we are here and we are supposed to handle the eukaryotes. People don't know that eukaryotes are different from eukaryotic cells. But I'm going to give you the roadmap. Okay. So we have that eukaryotes. What else? Then I'll give you some sort of assignment or research. You all know that there are classes where I go with assignment, there are those where I go with research, there are those where I go with both. So let's see how this lesson goes and we shall see what we shall have in the end. So these ones are supposed to be dusted, you should take that. And then I will brief you about the next stop. Okay? I'll brief you about the next stop. Okay, now the other time I told you about the prokaryotes. I told you prokaryotes are organisms that of course have cells. So the kind of cells they have are the prokaryotic cells. You understand? When they say prokaryotic cell, it is a cell of a prokaryote. So the prokaryote is an organism, then the cell is the prokaryotic cell. Are we together? So I want to be very fast here. I give you the brief introduction. Okay, what they call the preamble, the primary knowledge. So let me give you the primary about these ones. Okay, we are saying that the eukaryotic, okay, when they say eukaryotes, I've said here they mean the organisms. Yes, igi man. Man, I mean male and female, yeah? So that means you, the person, you are an eukaryote. Are we together? Then these ones have eukaryotic cells. Then, if I'm not mistaken, I remember the other time I asked what eukaryote means, and you people went and checked the internet and you told me about something like true. So you were like true nucleus, true. That's what you guys were saying. So you're not supposed to take that for granted, and I'm not repeating it. So you all are aware that we made research about the eukaryote part. I even remember teaching you how it is pronounced, and this was based on the fact that we have the presence of a true nucleus. Okay, very important. And I don't know if I have any student who is online and can really tell us what do I mean by a true nucleus. Yeah, why do we call that of prokaryotes untrue or false, and we call this one of eukaryotic cells a true one? What really makes the nucleus true? They're the things that we need to learn today. Do I have anyone who wants to begin the session with me? I'm seeing Esther's and Harp. I don't know. Okay, let me hear from Joshua, but please be fast.
I
Joshua, I've
think
asked you to unmute. Uh-huh.
a true nucleus means it has a nuclear membrane.
Thank you so much. Yeah, that one attended the class. So by having the true nucleus, we mean that there is presence of what we call the nuclear membrane, or which we gave another name. I remember telling you that it is nuclear what? Envelope. And remember telling you that the envelope is there to remind you of the actual envelope in daily life. It has two papers on sides, trying to communicate that even the nuclear membrane will be a double membrane. Okay? You shouldn't forget that. Then I remember telling you about prokaryotes, that they lack in one way or the other organelles that have a membrane or even a double membrane. Are we together? So the other thing um, that is so special about eukaryotes is having organelles that are membrane bound. Okay, so important. So we are going to see other factors in the notes we are going to look at, but have it in mind, these things, all those features that are so distinct about them, make them more complex. Okay, they make them more complex than the prokaryotes. Hope people are taking notes, hope people are listening and getting everything. We have just started. Let me hear from Esther. Esther, as if you want to say something, please say it. We proceed.
Uh, excuse me, sir. Good evening.
Yes.
I wanted to know, like, how much, I mean, for how long do this place, like, courses, online classes take?
You send me a message on WhatsApp. That's why I told you that to get in touch, you do that. But of course, holiday programs take holiday. Homeschooling, they take the term period. Accessing the drive, I told you the whole year. So anytime you want, you just access it. It's the one that has all these classes. I even thought the question is to do with content. Let me tell you this. When I start teaching, you just ask me content and I speak, because for me I be locked in. Okay, that's why I give that first period time. When I begin, we have begun. Okay, so those factors about eukaryotic cells, all those features that really make them distinct from others, and in the end, we have realized that they are the ones that make them more complex than prokaryotes. If you can recall, we said the prokaryotes were the simplest, earliest, and the most primitive, if you recall. Okay, now That introduction is just enough. So let's go through our notes. We now begin talking. Let me tell you this, especially those who will be listening to the recorded class. When I say something that I don't write, you're supposed to pause the video and write. And I want to give you a disclaimer at the beginning of the class that this topic, cell biology, is to do with a cell. In each and every topic you're going to cover in biology, a cell is involved. So it carries so many examples from other topics. Some of them I'll be telling you we hold on to wait for that topic. Others I'll be saying something small that can make you to understand a statement. But when I say those statements, you will appreciate them because they will have made sense, but you're going to forget immediately. So as a student, after taking the screenshot, you're supposed to note. You pause the video and note. You is attending a live class. You're supposed to watch this video twice. But for the second time, you watch it at a speed of times 2 so that you save time. After all, you attended it already. You're just attending it for the second time to take those notes. Things I said but didn't write because they're always high yield. Are we together? So before I even look at, so we are going to first have a look at the structure of the plant cell. We are going to look at the structure of the animal cell. And before we all do that, I think let's first look at the characteristics. Or before we even look at the characteristics that really make them distinct, yeah, I think they are these ones, the distinguishing features. Let's try to go to YouTube and try to watch some animations for the plant cell and animal cell. Because I told people to do that the other time, I doubt whether people did that. I think it was part of the research work. I don't know. So let's go to YouTube and people say something. Where is my YouTube? I think it's here. Okay, so we want to look at the animations. What was this? Oh, you see, I also use YouTube. You can see I was making research about the adult rhabdomyoma. Yeah, it's a pathology for the cardiac system. They are called rhabdomyoma cardiac tumors. Yeah, so I was making some research on echocardiography. There is some thesis that I'm organizing. So you see, even me, a doctor, I still use YouTube and watch some things to make my research so nice. But there is you, even when I try to use them in class to show you that they should be part of you, don't want to make research. Do you know the adult rhabdomyoma? Do you know the infertile one? Yes, but don't worry, you're also going to join me this side. And I also tutor you in pharmacology and pathology. So we shall be together till you get your final job. You never know, I may even give you connections of which hospital you may work in. Because some of us are too competent in the system. So we are going to look at animal cell. Actually, let's begin with plant cell animation, plant cell structure. I like 3D animations so much. 3D animations. Okay, let's see what they have for us. Rhabdomyoma. Okay, where is it? The secret world inside plant cells. I think this will work. Yeah. I think this will work. So let's watch it. So you keep quiet and listen. Let me hope I don't have students who get issues with the English of these words on YouTube. So you listen and let's see what the plant cell has. Then we also connect to the content. Yeah, we go.
Plant cell, similarly to an animal cell, contains a cell nucleus in which genetic material is stored in the form of DNA.
So I'm going to be like your DJ, but I don't even need to be a DJ. Don't you think the voice and the captions attached are just enough for you to watch and understand? Like they have said, plant cells, just like animal cells, they do what? They have a nucleus that is trying to enclose the genetic material, which is in the form of DNA, things like that. But it's just 2 minutes. So I think let's use the captions and the voice. The voice is a bit clear. You just watch, then we go to the animal cell, then we go to our notes and explain content. Okay? We go. There it is. You can see they are showing us green, communicating that we are going to talk about a plant. We go.
A plant cell, similarly to an animal cell, contains a cell nucleus, in which genetic material is stored in the form of DNA. Inside the cell nucleus, there is the nucleolus. It is a granular body built of protein and ribonucleic acid.
Yeah, sorry for putting it back. I was trying to improve the quality and increase the size of the screen. We go.
A plant cell, similarly to an animal cell, contains a cell nucleus, in which genetic material is stored in the form of DNA. Inside the cell nucleus, there is the nucleolus. It is a granular body built of protein and ribonucleic acid, RNA, in which, inter alia, ribosomes are produced. Ribosomes are organelles involved in the synthesis of proteins. Some of them are attached to the endoplasmic reticulum. Others are freely suspended in the cytoplasm. The cytoplasm is a liquid suspension filling the cell, which contains various organelles. One of them is the endoplasmic reticulum. It has the shape of a labyrinth surrounding the nucleus. One part of the ER is called rough, and the other smooth endoplasmic reticulum. The Golgi body is the cell repository. The substances produced in it are stored there and then transported to other parts.
But am I Uganda hearing the English? And are you getting it? Are you people following? Have you heard well about the protein synthesis? Have you heard about the ER, the endoplasmic reticulum? Have you heard of the rough and smooth? Have you heard those things? Now they are saying Golgi body. So you're supposed to listen to those things well as you're watching. Do you see the Golgi body, that yellow thing? Did you listen to things like the endoplasmic reticulum looks like a labyrinth? Did they tell you that it is either smooth or rough? Hope you're hearing those things, eh? So be serious. We go.
Of the cell. Among the more complex organelles are the mitochondria, in which cellular respiration takes place. Their number, size and shape vary depending on the cell type. Each plant cell is surrounded by a cell membrane. It acts as a barrier separating the cell from the external environment. A plant cell also has a number of unique features. The cell wall, which is composed of multiple cellulose layers, is a characteristic feature of.
Are you people really listening? Whatever the guy is saying, it is content. They have told you the plant cell is having a cell membrane. Did you listen to that? They told you that this cell membrane is a barrier, trying to hide it away from the external environment. They told you it has a lot of unique features, and the major one is the cell wall, and it's what they are talking about right now. Are you really listening? We go.
A plant cell. Its rigid structure serves as a frame that gives the cell a regular shape. The cell wall protects the cell against water loss, while allowing the flow of various substances from and into the cell. The plant cell can use solar energy as fuel for the production of the organic compounds it needs. This is possible.
Now, those things you have heard, that the plant cell can use solar energy for the production of organic compounds. It's what you guys call the plant cell using sunlight to photosynthesize. Because sunlight, of course, it is in line with solar energy and production of organic compounds. We shall see that under nutrition, that it is the photosynthesis. Are we together? They even told you that this cell wall is having some good shape, and it's the one that really does it all to give shape and rigidity and support to the plant cell, okay? And it even allows some movement of certain materials in and out of the what? Of the cell. Yeah, this video is really high yield. I'm even happy that it is ending.
Through chloroplasts found in the cell, which are also known as green bodies. These organelles contain chlorophyll. the green pigment enabling the conversion of solar energy into the energy of chemical bonds. Large vacuoles, whose surface often occupies almost the entire cell, are another unique feature of plant cells. Vacuoles can be like...
But you people don't just listen to the vacuole, look at it. They have told you that it is so big that its surface almost occupies the whole cell. And because of that, the nucleus is pushed to the periphery, and we shall see all that the nucleus will be found at the periphery. But then this one, the subvacuole, or the vacuole, cell vacuole, I'll tell you why it's called the subvacuole. It's going to take up almost its surface is taking up almost the whole cell interior. They told you about the chloroplasts. They also call them what? Green bodies. Why? They contain chlorophyll, and that chlorophyll is key in photosynthesis. They told you about the mitochondria, which are sites of what? Respiration. This video is really nice.
Likened to huge tanks filled with cell sap, containing mainly water and spare substances. These elements of the plant cell closely cooperate together, ensuring the proper functioning of the cell. They are found in all plant cells.
Now to okay.
Irrespective, these elements of the plant cell closely cooperate together, ensuring the proper functioning of the cell.
Now, to really confirm that you really people have been listening, I'm going to ask you certain questions in the chat, then you respond. What is that purple thing? I'm seeing some purple thing. What is its name in the chat? Tell me. I'm seeing the purple thing. What is that purple thing? You tell me in the chat. Because some of you are just good at watching and not understanding. What is that purple thing? Someone has said it, nucleus. Thank you so much, Vicky. I'm seeing that colorless thing. It's kind of gray, but it is very big. Down it is having two green bodies, then on sides on top it has some orange bodies, but for it is somehow glassy, somehow clear. What is that thing? What is that thing? Vacuole. Thank you so much. I'm seeing...
They are found in all plant cells.
Oh my God.
Irrespective, these elements of the plant cell closely cooperate together, ensuring the proper functioning of the cell.
Okay. Then the other thing, I'm seeing those green bodies. They are kind of piled up together. They are near that. They are just adjacent to that clear thing you've just called the vacuole. What are they? They are kind of yellow. What is that? What is that yellow thing I talked about? Someone has said chloroplasts are yellow. Really? Someone has said mitochondria. You're not easy. You people. Please, Ivan, leave the screen. Leave the screen alone. Okay. I think you can just look at all of them now. So you have the nucleus, and then around the nucleus, that purple thing, you can see it is the rough endoplasmic reticulum. You can see the chloroplast. You can see the vacuole, as you guys named it. The mitochondria, or the mitochondrion, is that one that is orange. So have it in mind, it is kind of oval-shaped. Even the chloroplast is kind of oval-shaped, but for it is green. Yeah? Then these yellow bodies, that is the Golgi apparatus. The moment you see yellow, you think about it. Okay? Then concerning the endoplasmic reticulum, it's one of the things I'm supposed to explain so much. But when you look at it here, I want you to see that it is a continuation of the nuclear membrane. Do you see the cell nucleus? That one surrounded by the purple labyrinth. So it has a membrane outside it. You all know the nucleus has the nuclear membrane. So do you see that these are the things that are coming off the rough endoplasmic reticulum, the smooth. Do you see that they are a continuation of the membrane that is surrounding the nucleus? So that is also very key. So the other things that come in, it is the cell wall, cell membrane. We are going to see the middle lamellae. We are going to see the plasmodesmata. We are going to have a look at a colored structure and also look at a structure from biological science, so that we relate the black and white and the colored, something that is very key. Okay, so they are the same things that that we are going to find in the animal cell, but have it in mind the animal cell is going to be somehow different. Do you see the shape of that plant cell? Is it regular or irregular? You just look at it. Look at the shape. We even mentioned it as they were talking about the cell wall. And these are things I'm not going to repeat when you reach the notes. I'm using this chance to talk about everything. So you should make sure you appreciate every statement. A statement that confuses you, raise up your hand. I choose you. You tell me to pardon and I will do. You tell me about the shape of that plant cell. Do you see it as regular or irregular? Someone is saying irregular. That is very bad. Don't you see that it is looking like a polygon? You look at it well. Don't you see that it is like a polygon? You count those sides and see. 1, 2, 3, 4, 5, 6. It's looking like a certain hexagon. Because for me, I'm counting about 6 sides. So it's looking like a 6-sided polygon. So the shape is regular. Is that okay? But we are going to see that of the animal cell being irregular. What is making this one of the plant cell regular? The cell wall. So that cell wall we talked about, it is very key. And I believe in the video you realize that what does this cell wall have? The cell wall has what they call cellulose. And we are going to see that there are other kinds of cell walls that have some other kind of substances. And at this very juncture, you'd want to inquire from members, what is that substance that we find in the cell walls of prokaryotes? I remember telling you that name and even giving you another name or the synonym to that name. What name is given? I want you to write it in the chat. You tell me the name of the substance, the chemical compound that comprises the cell wall of prokaryotic cells, and also tell me its synonym, because we are here to revise. Murrain, thank you so much. Peptidoglycan, thank you, people. Okay, so it's peptidoglycan or murrain, and here today we have learned that for the plant cell wall, it is cellulose. We are going to see something about other cell walls and what they contain, and you're going to be shocked. Okay, so I think I've said a lot about the plant cell. So let's also first maybe go back to the notes. We see our structure, then we come back and then watch the animal cell also. So I've been telling you that for the animal cell, it will not have the chloroplasts because animal cells are not supposed to photosynthesize. You're made up of animal cells because you belong to kingdom Animalia, so you're an animal. Do you really photosynthesize? No. Do you need chloroplasts? No. Okay, then you also don't have a cell wall, so you'll end up having a shape that is not as regular as that of a plant cell. So your shape of, or the shape of your cells shall be what? Irregular. Is that okay? Then the other thing I want to tell you about SAP. Yeah, just the word SAP. We are going to find it in our notes, but SAP is just anything that is transported in plant tissues. You just understand that when you hear the word SAP, these are facts of biology. SAP simply means anything, any material, any substance, any component, any compound that is transported in plant tissues. That's what they call SAP. So I want to ask you, shall we really have a SAP vacuum or a cell vacuum inside? Shall we really have a SAP vacuole inside the animal cell? Tell me in the chat. I've told you that SAP is anything, is a substance, is material, is a compound, is a component, is something that is transported in plant tissues. So if that is the definition of SAP, shall we really have a SAP vacuum in animal cells? Thank you so much. We shall not have. Yes, we are moving. I'm now getting the vibe slowly by slowly. That is good. So we shall not have it. Are you aware that it is the size of this SAP vacuum that pushes the cell nucleus of the plant cell to the periphery? Because for us, we are used to the cell nucleus being in the center. That's what we know. If you ask a primary kid or a kid of O level that where do we really find the nucleus, they will tell you at the center. But I want to shock you that the reason as to why it is not at the center for the plant cell, it is because of that subvacuole. Because the sap inside is having so many things, as we are going to see. So they make it so large. So its surface occupies almost the whole interior of the cell. And then it pushes that nucleus to the periphery. I remember teaching you periphery and center under coordination. Sorry, your Form 5s. I taught it to Form 6s. But the opposite of central is periphery. Okay? Opposite of center, peripheral. Opposite of central, periphery. Is that okay? So let's see what we have in the notes, and then we come back for the animal cell. Trust me, there is totally nothing in this topic if at all you understand the organelles. We are here to understand organelles and their function because we shall need them in the end. And the target is clear: eukaryotes. Provided we dust them, well and good. Plasma membrane, it's going to be yours. And I'm going to brief you on what you're really taking home. Okay? Now, look at this structure. It may not have all of them, but it really has enough work. Okay? So you can see the nucleus. So you're going to follow my red ink. We are having the nucleus here. Okay? Then inside, it has what we call the nucleolus. We are going to see what the nucleolus does. Okay? It has the endoplasmic reticulum. If you're very keen, you can see that it is a continuation of the membrane that is surrounding the nucleus. And it is either rough or smooth. And we are going to see what makes one rough and why is the other one smooth, okay? We have the ribosomes, and these ones we are talked about under prokaryotes. I told you they are just freely suspended in the cytoplasm. Is it okay? Then the mitochondrion, and I told you how this one is really identified. How do you identify the okay, have it in mind that we can have the chloroplast when it is oval, we can have the mitochondrion when it is oval. I'm not supposed to talk about their structures right now so much. Why? Because the structure of the chloroplast is supposed to be taught in details in the topic of nutrition when we teach photosynthesis, and the structure of the mitochondrion is supposed to be taught in details under respiration. You understand? Because the chloroplast is the site of photosynthesis and the mitochondrion is the site for respiration. So they are talked about that side. Are we together? How do you identify the mitochondrion? Both of them have double membranes. The one of the chloroplast, it is just like the outer one. So for it, it just comes, they're just in line like that. The way you see it here, they're just in line. Are we together? But that one of the mitochondrion, the inner one is folded. Yeah, the inner membrane is what? Is folded. And it forms what is the name of those inner foldings? And let me see people who read textbooks. Yeah, the diagrams are a bit sketchy, but the good thing we have our mitochondrion here. You can see the inner membrane being folded into those structures. How are they called? Christy, thank you so much. That is the name. Someone is like, the image is not clear. I don't know why. Is it their device or true the images are not clear? I thought they are, but I've tried to expand everything. So you can see our Golgi complex. It can be called the Golgi body. It can be called the Golgi apparatus. Yeah? Hope we are together there. So you can see this vacuum. It is large and central. Hence it ends up pushing our nucleus to the periphery. Are we together? So we can see something here about the membranes that are involved. So do you see this very outer one? It's our what? Cellulose cell wall. It is the cell wall, but we have said it is composed of a carbohydrate known as cellulose. So have it in mind, cellulose is a carbohydrate. And you cannot forget that. You know glucose. Most carbohydrates always end in that. You know glucose, you know maltose, you know lactose, now cellulose. What really stops it from being a carbohydrate? So it's also a carbohydrate. Okay? So in the future, as we progress, you even learn that it is a polysaccharide. Okay? Just because carbohydrates are not on your syllabus. I don't know why that was done. but that's not our concern as of now. So the plasma membrane is inner to the cell wall. Hope you saw that in the video, that the cell wall is the outermost. Then after it, as we move into the inside, we shall meet the plasma membrane. So the plasma membrane has other names. They can call it the cell membrane. They can call it the cell surface what? Membrane. So cell membrane, or they can call it the cell surface membrane. All those are names for the plasma membrane. It is very okay. Are we together? So I think we have said some good work about the plant cell, and we've tried to exposit it in one way or the other. But when we check the outer structure in biological science, we are going to see more deeper structures, and I'm also going to be saying something small on them. So as someone who will be listening to a recorded class, you're supposed to stop the video and even first write this one in full. Otherwise, at the end of this class, you may not be in position to name that one was cell surface membrane. So you please be very technical as you're attending the classes of biology. Is it okay? Now, that is the plant cell. If you have any questions, you ask. Now, let's now try to have a look at. So you people, in case we watch a video and in your free time you'd want to really get in touch with the video again, look at the title of the video so that when you go back, you watch it. So you can see that here I'm seeing the YouTube channel is called Austin Visuals 3D Animation Studio. So you also note it down. In case you're looking for something, you do the same thing. Yeah? So I'm going to come here and say animal cell, so that we also see if we shall get animal cell structure. It has been called Austin Visuals. So I can also come here and add on Austin Visuals. Yeah. Let's pray that he also has one. That is human cell. I think we should watch the human cell. You're not supposed to watch a very long video. It's supposed to be very easy. And as you're watching, you're supposed to choose a video that is having so many views. That means it is a very good one. Inner cell life. I think we should watch this one. It is just 1 minute. Yeah, let's just watch and see. Maybe it can help. So this is now the animal cell. Don't worry, it is kind of delaying
Animal
because
cells have irregular shapes.
okay. This is an Indian. The moment you hear irregular.
Animal cells have irregular shapes. They contain a nucleus, cell membrane, cytoplasm, mitochondria, endoplasmic reticulum, Golgi bodies, centrosomes and ribosomes. Mitochondria are known as the powerhouses of the cell. Endoplasmic reticulum,
Now I
Golgi
expect
bodies,
now I expect people to take a screenshot for that thing, because it is a very nice structure. So do you see the ribosomes? Do you see those things of centrosomes? Those are things you're going to look at them. You're going to see them. But concentrate on the mitochondrion. Do you see that it is oval? Do you see the irregular foldings inside? Do you see the nucleus? Do you see that inside there is what they call the nucleolus? Yeah. Do you see the endoplasmic reticulum? Do you see as a continuation of the membrane around the nucleus, the nuclear envelope or the nuclear membrane? Do you see the cytoplasm, which is just inside the plasma membrane? These are the things of lysosomes. We are going to see them, okay? Do you see the cell membrane? Do you see that we don't have a cell wall? Do you see that this structure is irregular? It's not like the other one of the plant cell that was regular. Are you seeing all those things? So for me, I just believe that on the structure we have on the screen, it is only the lysosome that you've had for the first time. And you're also going to learn it today. What is it all about? Things like that. So let's listen to the guy. We complete, then we go to the next thing.
Centrosomes and ribosomes. Mitochondria are known as the powerhouses of the cell. They contain enzymes for aerobic respiration, and this is where most energy is released from respiration. The nucleus contains genetic material. This controls the cell's activities. Endoplasmic reticulum, fine network of interconnected tubules, a reticulum, hence the name endoplasmic.
Have you heard that we are having the mitochondrion, that is the powerhouse of the cell, that it is having so many enzymes that are responsible for strictly and strictly aerobic respiration, but we shall see all that. It's having the nucleus, and inside the nucleus we are having the what? The genetic material. But we said all that what makes the nucleus or of the eukaryotes to really be true, it's because it is having a double membrane around, and even inside we have the DNA material. It comes and it encloses the DNA material. We are going to also see that in the development of the eukaryotic cells. I told you cell biology as a topic, it's having so many concepts. You'll be hearing so many things and you may even burn out. But I advise you to just stay calm. You just make sure each and every statement being said, it makes sense. Other things, you see them after the class. Is it okay? So, let's go.
Reticulum ea. Microtubules are polymers of tubulin that form part of the cytoskeleton and provide structure and shape to eukaryotic cells. The cytoplasm is a jelly-like material that contains nutrients, salts, and other membrane-bound organelles. The cell membrane controls the movement of substances that go in and out of the cell. Ribosomes are small organelles found in the cytoplasm where protein synthesis occurs. The Golgi apparatus, also known as the Golgi complex.
Yeah, just because this person is going so fast, but he's speaking sense. The ribosomes, they are just also in the cytoplasm, and just like those of the prokaryotes, they are responsible for protein synthesis. Okay, they have told you about the Golgi body. They have told you about the endoplasmic reticulum. But first, we are going to see all that in details. I just want you to look at how they look like here. Then later on, when we begin content, you really connect. But I want you to look at that endoplasmic reticulum very well. Do you see that it is having so many cavities that are connected together? Those cavities are so important. Do people see the cavities? This is what I mean. Anyway, I will show you. Just make sure you have screenshots to the structure, so that when we go now to the black and white structure in biological science BS, you can really recall. Yeah? You connect the color to the black and white. Content will really come out. So this person is concentrating on the structure and function, but me, I was interested on structure. So I think I can stop the video there. You can go and watch as so many videos as you can. So I think straight away, we can now go back to the content. Now, before we go to the distinct structures, or the structures that really make the eukaryotic cells distinct or different from others, I want us to first also look at the colored structure for the animal cell. So this is it. Yeah, we are going to see that of biological science. So let's first pick out the common ones. The very first one is the plasma membrane. Here we don't have a cell membrane. That's how our structure is not regular, because the cell membrane is the one that puts up those situations of having a very nice shape. Okay, so here the shape is irregular. Let's first take the common ones. You know the ribosomes. I'll put arrows on them. They're just small things suspended in the cytoplasm. You all know the nucleus, and inside the nucleus there is that dark body called the nucleolus. Is it okay? You all know the Golgi apparatus. Let's first take the common ones. The cytoplasm, you also know it. Are we together? Now you can see that yes, we have a vacuole in the animal cell, but it cannot be called the sap vacuole, and for it cannot be so large. Yeah. But that of the plant cell is the subvacuole, and for it is large. Is it okay? Then you know that endoplasmic reticulum, we said it is either rough or smooth. Are we together? Yeah. Now, These other things of microtubules, microfilaments, we are going to see them together. Intermediate filament, that one we are going to see it as we talk about the cytoskeleton. So have it in mind that these things are there in the notes. Don't worry. After all, we shall later on take screenshots. So you just listen and understand. Yeah? The cell has what we call the cytoskeleton. Remember, cyto is for cell. I told you that in lesson 1. So if I say cytoskeleton, I mean a skeleton that is supporting the cell. So that cytoskeleton is made up of 3 things. That is the microtubules, the microfilament, and the intermediate filament. So we shall see them in details, don't worry. The mitochondria, this one we know, but it is plural, so they are supposed to be 2. When it's 1, it's mitochondrion. Are we together? So there are these other things that people are not getting well, but I'm going to explain them. Are you seeing like this peroxisome? That one, it is just a name. They are also called microbodies, but things are there in the notes. We shall meet them, yeah? So they are also what they call peroxisomes, but they are also called microbodies. They are just inside there, the cytoplasm. So have it in mind, the cytoplasm is just there to accommodate these organelles. So when you see a new name, just appreciate, okay? But I will tell you why it's called the peroxisome, and the name shall be because of this. In the future, or as you progress, you will realize that most of the organelles of the cell have this word, some, centrosome, lysosome, ribosome. Are you listening? Peroxisome. And you will know why. Even if you just go to Google and ask it, why do most, you understand, organelles of the cell have that last word some? They will tell you and you will learn new work. So as I explain why it's called a peroxisome, I will concentrate on peroxy and you will appreciate. Then also as I explain why it's called a lysosome, or as you recall why it's called a lysosome, I will talk about the lyso, because some is not so special. Even the centrosome, I'll explain about what? Centro alone. But ribosomes, you already know. Okay? You already know that why they are called ribo. Ribo is because of RNA. And you learn that in nucleic acids under inheritance and evolution, the 8th topic in the curriculum. Are we together? It's actually the 7th. I beg your pardon. So do you see? You're seeing something to do with the chromatin. Now you're supposed to understand chromatin in the topic of cell division. But I told you, when you hear things like chromosome, chromatin, chromatid, as of now, take them as DNA. Don't overthink. So when you see chromatin, for you just understand that is genetic material, that is DNA. For now, don't overthink. But when you reach our topic of cell division, I will break for you down what a chromatin is, what a chromatid is, what a chromosome is, and how they relate. What homologous chromosomes are, what sister chromatids are, things like that. You will appreciate. But for now, when you see chromatin, chromatid, chromosome, that is genetic material, that is DNA. Don't overthink. Okay? Is there any structure I've not talked about? The Golgi vesicle. You people, when you see something small like that, it is a vesicle in this thing, in this topic. So these are just, when they say Golgi vesicles, they are meaning vesicles that are just simple structures like these ones that are coming off the Golgi apparatus. Don't even overthink. Secretory vesicle. That means when you see vesicle, it is something like that, having something inside. When they say secretory, that means it is really giving us a certain secretion. You people, I think you've heard of things like secreting HCl, secreting gastric juice, secreting. You've heard of those things so much in digestion. But even if I don't use digestion, which thing have you had that is that has ever been secreted? I think you've heard of secreting enzymes. You've heard of secreting hormones. Haven't you heard of those things? So if at all they are to secrete a hormone, maybe the hormone comes out as a secretory vesicle first, then maybe the secretory vesicle bursts and the hormone goes out. Do you understand? So when you come across those things, don't even be surprised. They are nothing. They shouldn't overwhelm your mind that you're learning a lot of new things. Just calm down, breathe in and out, break down the word. Vesicle, you know what it is? Secretory, because it is giving us a secretion, which could be a hormone, which could be an enzyme, which could be a juice, digestive juice, saliva, things like that. Okay, so I think most of the things have now been talked about. Okay, what we've not talked about is this centrosome. But what I want you to appreciate about the centrosome as of now, I want you to appreciate that there are two things. Are you seeing this centrosome? There are two of them. One is here, another one is here. They are just next to each other, and they are next to each other at right angles. Just because this is a luxurious diagram. But for you, as you're drawing them, you're supposed to just do this. You draw one like that, then you also draw the other like that. You don't waste time, just like that. That's why they tell you that they are always occurring in pairs and at an angle of 90. You understand? Occur in pairs at right angles. Occur in pairs at an angle of 90. You get? So most of the things have been talked about. So in case any organelle has not been clear, you just raise up your hand and tell me, doctor, I'm really not understanding that. And trust me, those organelles come back in all topics you're going to cover. Father, mother die, they will come back. So if you don't understand them here, they will give you a time in the future. You're going to see in the functions of like of the lysosome. We are going to talk about a sperm. But a sperm is in reproduction, even when it's not in your syllabus, but you meet it somewhere. We are going to talk about things like the cytoskeleton. It's going to come up in so many things. The mitochondria, if you don't understand it, it's going to give you a time in respiration. The chloroplast, it will come back in nutrition. The Golgi apparatus, it will come back in homeostasis. It will come back in hormonal coordination. Okay, so make sure you appreciate. That's why I'm giving the introduction a lot of time, because I know what it means if a student cannot really tell anything about any organelle. They put you there and tell you, say something about the microtubule, but you just keep quiet. You can't even say something that it is part of the cytoskeleton that supports the cell. That is kinda absurd. So that is it. Now, immediately after having a look at the colored structures, now let's look at the structures of black and white from biological science. So this is from biological science. Those who have BS, you can check under cytology, of course. Okay, then we are also going to see that one of the animal cell. Trust me, when you will look at these organelles and see wherever they are, we see each and everything about them. My dear, the notes we are going to just be speaking and going through everything chap. And in no time we shall leave this place. Are we together? So we are still on the structure. It is the key target for the class. So this is the plant cell. Do you see that it is regular? Even here it's having 6 sides. So it is the hexagon, though it is kind of funny, but it is regular. The sides are 6, and it's also having 6 vertices. So it's just perfect. Are we together? So I told you that the outermost we shall have, I think let me use orange. The outermost we have is the cell wall, that one. Hope orange is very clear. I don't know. Let me try blue and see. The outermost is the cell wall. After the cell wall, we expect what we call the plasma membrane. And I told you it can be called the cell membrane. It can be called the cell surface what? Membrane. Now, before the cell wall, you can have something they call the medulla mela. In plural, it is medulla meli. You all know that in most of the terminologies of biology, we end with that E to mean plural. Are we together? Now, after the plasma membrane, what the plasma membrane encloses, that liquid, is what we call the cytoplasm. Are you seeing? Hope you're following my arrows. Cytoplasm. So that whole thing. Now, this cytoplasm is having a lot of things. I want you to listen carefully. After the cell wall, we have the plasma membrane. Then the plasma membrane encloses what we call the what? The cytoplasm. Now, within this cytoplasm, we are having so many things suspended in it. And I want to tell you today, something that they will never tell you. Okay, they will tell you at some time, but it is something that people never understand, or something that is underrated. So this cytoplasm is having 3 things, and they are so important. And when you go back to school, at least tell a friend you really care so much about. Tell them, did you know that the cytoplasm is composed of 3 things? And if I have a student who can mention those 3 things, I'm going to give you 100,000 right now. That's if you raise up your hand immediately in less than 10 seconds. Otherwise, if they go beyond 10 seconds, that means you've copped. So these are, okay, I'm going to choose wisdom. Carrots. 3 things. Okay. So let me hear from wisdom. Let me hope, wisdom, you've not raised up your hand so that you can buy time as you copy. I will tell whether you've copied, because I'm going to ask you more complex questions about what you are going to submit. Yes, wisdom, you speak. I asked you to unmute. What are those 3 things that make up the cytoplasm? Yes, please.
It's made out of the simple organelles like
Yeah, that is one. Then the next thing.
food nutrients.
That is not right. Okay, you had said food nutrients. Okay, Ken Wisdom, you give me the 3rd thing. You've said food nutrients. What else? I think he's now off. You unmute and tell me the 3rd thing. You've said organelles, you've said food nutrients, and what's the 3rd thing?
That is it, sir.
Okay, good try. 2 out of 3. Okay, he has tried, though he has not brought out the one of food nutrients well. Now, the cytoplasm is divided into 2 things. It's having what they call the fluidy part. Most people think the cytoplasm is the fluid part of the cell. You're lying. That's a total lie. So the cytoplasm has the fluid part. So if they don't call it the fluid part, they can call it the soluble part. Are we together? Now, it has a name. Then, of course, it has the non-fluid parts. And the non-fluid parts, they are going to be 2, and you're going to learn them. So the fluidy part of the cytoplasm, which is the actual fluid part of the cell, is what they call the cytosol. You read FA and you will find it. And I know there are people who are hearing cytosol for the first time. Okay? And then cytosol has other names. It can be called the ground substance. The ground what? Substance. Then rarely they can call it the matrix, but that is rare. The most common ones, cytosol, then ground substance. It is the fluid part of the cytoplasm, which is the actual fluid part of the what? Of the cell. Now, other things inside the cytoplasm, they are non-fluid. I will not say they are solid, I will not say they are gaseous, but have it in mind they are non-what? They are non-fluid. And those are the organelles. We are going to define organelles today, and I told you that organelles are simply distinct structures of the cell. You saw that the ribosome was somewhere, even the lysosome was on its own. The mitochondria was somewhere very far, even the chloroplast was the other side. So you realize they are distinct. They have particular structures, they have particular functions. That's what they call an organelle. Are we together? It is distinct, meaning that you can never find the chloroplast together with the mitochondria. No, they are separate, so they are distinct. They have a particular structure. You saw that a mitochondrion differs in one way or the other from the chloroplast in a way of the orientation, organization of the inner membrane. So their structure is particular. Even their functions are particular because why? The chloroplast does photosynthesis, the mitochondrion does aerobic what? Respiration. Then the other non-fluid thing are what they call the cytoplasmic inclusions. Okay? They are the cytoplasmic inclusions. I told you about these ones in the lesson of prokaryotes. So the organelles, have it in mind, they are living. They are living. Yes. Then have it in mind, these inclusions, it is the opposite. They are non-what? They are non-living. So these inclusions, they are normally those towers. Yeah. You all know. Now let me teach you something. I'll tell you about these inclusions. Okay, these inclusions are those granules that we meet. You all know that plant cells really photosynthesize. And when they photosynthesize, they give you starch. And there is need to store this starch. So in a plant cell, this starch shall be stored in what we call starch granules. Are we together? There is a lot of metabolism inside a cell that occurs. And we can have conversions. We can form lipids. So these lipids are also supposed to be stored in what we call lipid droplets. We shall see some other word for droplets. Can be called lipid globules. Anything. Metabolism happens and it involves carbohydrates. And an example of a carbohydrate that we have that is so good to man is glycogen. Just because it was removed from your syllabus, but you could look at the properties of glycogen and really appreciate it. It is really a very good store of carbohydrates. That's why the animal cell will prefer storing its carbohydrates as glycogen. And glycogen is supposed to be stored in glycogen granules. So those things of starch granules, those things of lipid droplets, those things of glycogen granules, those are what we call inclusions, and they are non-living. They are just stores. They are just stores. But then you look at a chloroplast, a lot of metabolism is happening inside, so it is living. It is using ATP. How do you expect it not to be living? Hope we are together, people. So I want you to be that kind of student who can break down a cytoplasm as a student of SSA. You say it has the fluid and non-fluid. The non-fluid has the living and non-living. The fluid can be called the soluble one. It is cytosol, ground substance, or rarely matrix. Organelles you mentioned, some even define it. Tell them why they say distinct. Tell them why they say particular structure. Tell them why they say particular function. And you'll be a very good biology student. You will write very good work. And you will make a very good person in the future. Okay? People told me that they read and forget, but that is very simple to really control. And I always tell you in the channel, just because people don't listen. So you're supposed to remind me after this class. I tell you about how best you can really make this content to be part of you on and on. But it requires full-time reading. So if you're this student who is also good at scrolling on Instagram and TikTok, whatever I'll tell you may not work for you because those things are addictive. Even me, some years back, I got addicted to some sort of scrolling on Instagram, and it really affected my research. So it is really bad, and it is always hard for someone to really have control over it, because sometimes you can be like, let me read 2 pages, I can't scroll only 2 videos, but you end up reading half a page and you scroll over 100 times. So you should, whatever I will tell you is not really compatible with you scrolling. You understand? So that is it. Now, we can go back to our notes and see. That is the cytoplasm. So you now know. These things, are you seeing this is the chloroplast? So these things of chloroplast, envelope, and granum, these ones don't concentrate on them right now. I told you that the chloroplast is going to be taught in detail when we reach nutrition. Is it okay? Yeah. Which other one? Free ribosomes, these ones you know. Endoplasmic reticulum. Now let me tell you also about the endoplasmic reticulum so that we also now take that one as a done deal, yeah? Because I'm tired of over-promising you. Now, you people, the nucleus. Remember we said it is surrounded by the nuclear envelope. Now let me shock you. This is the nucleus. Because I believe most people don't know how the nucleus really looks like. People think it is just a circle. But it is not, my dearest. Yes, it's supposed to be circular. So you forgive my art, but at least I've tried. But have it in mind, whatever I'm drawing is circular. So while you're seeing this one as a double membrane, we want to bring out the other thing of it being called the nuclear envelope. And I told you why envelope, to bring it out that it is what? A double membrane. So this one is called the nuclear envelope. Now, there is okay, what do you think are those spaces I've left within? Do you think they really have a name? Do you think they have a name? So we have said that this one is the nuclear envelope. So this thing is what they call the nuclear pore. Maybe I can name it here. N is for nuclear, then pore. So inside, I told you we have that dark body. Yeah? That nucleolus of ours. It is somewhere. Not at the center, but somewhere at the periphery. The nucleolus. Very important. It plays a very good role, and we are also going to see it here. Nucleolus. Is it okay? Then, this is what I've been always telling you, that the endoplasmic reticulum you're seeing as a continuation of the nuclear envelope or the nuclear membrane, the endoplasmic reticulum you've been seeing is actually a continuation of the what? Nuclear envelope or nuclear membrane. This is what I've been meaning. It comes off like this. But remember, it is also having connections. Are we together? So it can come off like that. Know that you're getting it. Like that. And it continues. It can come off. It can also be curved. It is very okay. That. You see that? You see? Things like that. You forgive my sketches, but I want you to get the concept because I believe you watched so good work and I want to be a bit fast when I'm showing you this. So that's what I mean by the continuation. These connection points, very high yield. Okay, and you can also see that even the reticular, they are also connected in one way or the other. So these cavities, I want you to learn. Someone said that those enfoldings of the inner membrane of the mitochondria, they are called cristae, which was very correct. But that was for the mitochondria. Someone said they are called cristae. That is very good. But then even these cavities for the reticular, they also have a name and they are called the cisternae. I used to find issues with students confusing cisternae and cristae, but hope you get the point, that those inner enfoldings for the inner membrane of the mitochondrion, they are called cristae. But then these cavities for the reticular coming off or being continuous with the nuclear envelope, for them they are called cisternae, and they of course interconnected. So you can see that they are also what? Interconnected. Now this is where the difference really comes in, and I want you to listen carefully. We have the reticulum that is going to come off with some ribosomes on its structure like that. Are you seeing those ones? There are going to be so many. Then we are going to have the other one that is clean and plain. It has nothing on it. Are we together? This one is just clean, nothing on it at all. So this one that is going to be having ribosomes, I've told you that these ones are ribosomes. Don't think it is anything else. They are what? Ribosomes. Then this one is just clean. But remember, all of them are continuous with the nuclear envelope. So they are the endoplasmic reticulum. And have it in mind, it is abbreviated as ER. So I'll be using ER to mean endoplasmic reticulum. endoplasmic within the cytoplasm. Why reticulum? Because it is made up of these cisternae that are interconnected. If you want to say only one, it is cisterna. So cisternae is actually plural. Are we together? So this one that is having ribosomes on it, look at it as the rough. So you seeing that R on ribosomes, this one makes it to be called the rough ER. So you have two options. You either be like the ribosomes maybe make it rough, or you can say rough, and this first R is just there to remind you that it has ribosomes on it. So important. Then this one that has totally nothing, it is just smooth and very clean. It's what they call the smooth ER. I've told you YE, I've told you what YR. Are we together? So the content we are going to explain, make sure you know this sketchy content about the endoplasmic reticulum, because it is going to help us so much in the future. Are we together? Now, I think we can go back. If I leave you, please ask. So when you look at this rough endoplasmic reticulum here in red, do you realize it is having some things on it like pimples, like rashes? So those are ribosomes on it. Yeah, very important. And then when we look for the smooth one, we are going to find it somewhere, and for it, it will not have anything. It is here. Though the word smooth was written wrongly in the biological science, but do you see it? It has nothing on it. But the rough one had things on it. Are we together? So I don't expect that one to be a problem again. And they are trying to show you that this one is very fine. Someone may be like, but it is not continuous with the with the nuclear membrane, but it is even here. Are you seeing, oh, that one is the rough. Then this one, the smooth. Are you seeing, oh, this? This one is still the smooth. Have it in mind, it is always continuous with what? With the nuclear membrane. Now, this is the mitochondrion. And you see its inner membrane having folds. That means even without telling you this one is what? Mitochondrion. Without telling you this one is what? Mitochondrion. Then I told you the chloroplast, for it is just having its oval as normal. The inner membrane is just like the outer one. So when you see something like this chloroplast, even this is a chloroplast without even anyone telling you. Even this one is a chloroplast without anyone even telling you even a simple hint, because you know how things look like. Okay, now someone may ask, then how will the Golgi body look like? The Golgi body will have things that are piled together. That's how it will look like. It will kind of try to resemble the endoplasmic reticulum, but for it, it is on its own. It is somewhere very far. I want you to look at it like this thing. That's how the Golgi body looks like. Then vesicles try to move off it. Those are what they call the Golgi vesicles, and we saw them together. Okay, then it's also having those cavities with the interconnections. I don't know what I'm going to do here. See those things? So the vesicles go off like that. I don't know why, but for some reason, they didn't want you guys to learn how these organelles really operate. They just wanted you people to know their functions just without really concentrating on how they are really doing their things. But I'm going to be giving you hints, and I also advise you to go where a function has really disturbed you. You can make research. You can find out why is this like this and like this. So I've told you I'll just draw my structures in a funny way just to get done with all this. So I've told you the vesicles go off like that. So this is supposed to be a connection, something like that. So if we just look at the Golgi apparatus, or the Golgi body, or the Golgi complex, as things trying to pile up like this, like this, like this, and then the Golgi vesicles are what? Are going off. That's how they will show it to you. Here I'm just trying to expand it and show you the real detail. So these ones are also still cavities that are joined together. So don't hesitate to call these ones the cistani. This is very okay. Then these ones are Golgi vesicles. They just pinch off here. The moment it reaches here as a vesicle, it pinches off, then it goes off. Pinches what? Pinches off. Then it becomes vesicle. Then I'll tell you more as you read through the functions. Right now, appreciate the funny structures. When you reach the functions, I'm going to be coming back. Then I tell you that in case it pinches off, it can become this because of this. It can become a lysosome because of this. It can become this and this because of this. Are we together? And you're going to be surprised that there are some organelles like lysosomes and peroxisomes that come from the what? The Golgi apparatus. So it is very key. But let's first reach it, then we come back to this diagram of ours and we say more about it. Is it okay? But why I'm showing you this, I want us to be in position to identify it on the plant cell so that we are not really disturbed. Yeah, we don't really need to be gamblers when it comes to content. So when you see something like this in green as a biology student, you should be like, oh, I'm seeing some things that are piled on top on each other, and on top I'm seeing things that are going off. You'll be like, this one is the Golgi complex, Golgi apparatus, Golgi body. And in the plant cell, there are special Golgi bodies that are called dictyosomes. Yeah, that is just high yield. It's not high yield, but it is a fact. Some special Golgi bodies in strictly plant cells that are called dictyosomes. Are we together? Then, even without anyone telling you anything, you can really tell that this one is also what? A Golgi body. Why? There are those cisternae that are piled on top of each other, and you can see vesicles pinching off from the ends. Is that really hard? Not at all. So I told you about the microfilaments, and I told you I will tell you more. I told you about the microtubules. Like, they are also here. Do you see them? Then I also told you about the intermediate filaments. I think these people didn't show them here. But you know that those ones simply make up what they call what? The cytoskeleton. And as you hear cytoskeleton, it is a skeleton to support the cell. Okay. Then the other thing is concerning the nucleus. I told you about the nucleolus, that one you know. I told you about the nuclear power, that one you know. I told you about the nuclear envelope. You also know that. Now, the genetic material, which we call the chromatin, is divided. Yes, and you should also learn this because you may meet it somewhere. And I'm also going to be forced to just say something small about it. So chromatin can also be divided in one way or the other. It has the light one. Are we together? Then it has the dark one. Yeah? By dark, I mean this one is more clear. Eh? Then, of course, light, the one that is light may not be so clear enough. Are we together? So, the genetic material can organize itself in two ways. It can either act as a ring or it can organize itself somehow circularly. But don't say that this is linear. No. According to what you see under a microscope, you'll be seeing the genetic material, but you'll be seeing part of it, they are lines. Another part, they are somehow like circles. Do you understand? So they call this a certain form of chromatin material. Even this one is called a certain form of chromatin material. Now you tell me, of the two, which one do you think is more visible? The one that is more clear, the one that will be darker. Which one do you think is the one? Do you think it's the one that is remaining as like a thread, or this one that is folding into something circular? Which one do you think will be more clear or darker? You just tell me in the chat what you think according to English. Don't overthink. Which one do you think will be more visible? In actual sense, what are you really seeing? For you, when you look at those two, the thread and the circular one, which one do you see easily and more clear? You people are not easy. You're saying the thread is more visible than the circular one. The thread, you may not even realize that it is something serious, but when you see something folding itself into a complete shape, you may be like, maybe this one really means something. So for me, I think this one is actually more clear or darker. You understand? So this one that is more clear or darker, it's what they call, all of them are chromatin. Yeah? So we have the euchromatin and the heterochromatin. Yeah. So the one that is more darker, you can see it is the hetero, the one that is trying to be somehow like that. Yeah? So it is the heterochromatin. Remember, we got this from biological science, which is your reference textbook. Eh? So whatever you see there, you cannot take it for granted. So this is euchromatin. So important. You can find any way you want to really recall it. But I've told you, heterochromatin, euchromatin. If you think that is very hard, you just say chromatin. If chromatin is hard, you just say genetic material. Genetic material is hard, simply say DNA. Okay? So, and all those things really make up what they call the what? The nucleus. And I think I've talked about whatever structure is here. Now, what is fun is that this thing you're seeing in the center, I told you it is the cell sap, or you can call it the sap vacuole if you want. But what is inside is the cell sap. And I told you what sap is. But its definition is even part of these notes. Let me tell you, most of the things we are going to meet down, I have said them all. So we are going just to be going through notes chap. You just need to rewatch the class, sit down, make notes, memorize, things of the sort. Okay? Now, the membrane that is surrounding the sap vacuole is called tonoplast. It is called what? Tonoplast. Now, the combination of the cell sap inside the vacuole and the tonoplast, which is like a membrane surrounding the vacuole, they make, okay, the vacuole at once. Eh? But have it in mind, the membrane that surrounds the vacuole, it is the tonoplast. Or the membrane that surrounds the cell sap makes the sap vacuole or the vacuole. Are we together? And that membrane that covers the vacuole is called the tonoplast. Are we together? So I think every structure has been talked about. And if there are any questions, you ask. But that is the plant cell for you. Okay? Now we go to the animal cell. Okay? And trust me, things here are the same. Just because the animal cell is kinda irregular. The shape is irregular. But the lysosome, ribosome, microtubule, I said. These ones, we said they are together and they are at angles of 90, and here it is very clear. You see, we have the heterochromatin here and the euchromatin, which is just chromatin. We have the nuclear pores, nuclear envelope, nucleolus, and all of them are making what they call the what? The nucleus. Hope we know that. Do you see the Golgi apparatus? Do you see how it is looking like? Do you see the Golgi vesicles trying to pinch off? Do you see a Golgi vesicle trying to become a secretory one? So that means if the Golgi vesicle gets a digestive juice inside, or if it's having a digestive juice inside, it becomes the secretory vesicle. Then it will leave the cell by exocytosis. When you see exo, you think of exit. When you see cyto, you think of a cell. And when you see cysis, like cis, it is a process. So it is a process of something exiting the cell. What is exiting the cell? The secretory product that is inside this secretory vesicle, which was just an origin, or whose origin is a Golgi vesicle, which Golgi vesicle pinches from one of the cisternae of the Golgi apparatus. Are we together? Microfilaments, we all know them, and I'll tell you why they are called actin filaments when I reach that part of the cytoskeleton. Just don't worry about that. Smooth endoplasmic reticulum, simply because it has nothing like pimples on it. It is just smooth. The pimples are the ribosomes. Are we together? You can see the rough here. Do you see the pimples on it, the ribosomes? They have even added it here. The mitochondrion is here. Do you see the inner folded membrane? The plasma membrane is all we have. We don't have a cell wall here. Are we together? The cytoplasm. Do you know its 3 divisions clearly? Which one I've not talked about? We have what we call the microvilli. Everyone knows that microvilli are responsible for absorption. And when it is one, it is called the microvillus. Do I really have anyone here who doesn't know that microvilli are really responsible for absorption? I don't think so. So we have what we call transport across a cell, or movement in and out of a cell, and we have what they call pinocytosis. So pinocytosis deals with drinking. When you read FA, they will tell you that pinocytosis is what they call cell drinking. But I don't know why that was removed from your curriculum, but it is something so important. So when you see something like pinocytic vesicle, the cell is drinking. So they are trying to show you that pinocytic vessel forming, the cell first forms that kind folding, then it takes up the drink. When it takes up the drink, it now closes and the vesicle enters inside. Are we together? Then if it is phagocytosis, it is cell eating. The cell is eating. So it also forms that car pocket. Then the solid food enters, then it closes, then it takes up the solid food. If it is pinocytosis, we are drinking. It also makes that car pocket, the fluid enters, it drinks, it closes, the vesicle enters. Then we shall see the fate of that vesicle later. We are going to see who really breaks those things that are inside. I remember in the topic of chemicals of life underwater, I told you about hydrolysis. I told you when you see the word lysis or lysis somewhere, that is breakdown. And here we have the lysosome. So the lysosome is responsible for breaking whatsoever is supposed to be broken inside the cell. And we shall also see that the ribosome has the special things that do the breaking. Are we together? So I think you have also appreciated the outer structure of the animal cell and plant cell, both the BS black and white and the colored, and also the YouTube videos. And that is let me tell you, even if I end the class here and I just give you these beautiful notes, you can read and understand each and everything, unless you read with bias. But almost everything has been said. Okay, now I want you to breathe in and out. You rest a bit as you tell me in the chat that we are really still together and we should really proceed. But otherwise, we have moved. That's what I can say. 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 Now, there are things that we call the protoplasm and protoplast. We have those things in the notes, but I always want to write them as I explain, but I may waste some time. The good thing I try to do for you, those things of the cytoplasm, even these things, my dear, these things, my people, these things that we put, cytoplasm fluid, part non-fluid, we have them in the notes, but I just always enjoy writing them so that you see how I come up with things and you get that vitamin of really content reproduction, like bringing back the content to life. But for protoplasm and protoplast, I think we are going to just find that in the notes. Then I say something about them. Think, yeah. So, are we really good? Should we now proceed? You first breathe in and out and at least rest cough for a minute. Because now we are going to read as I speak. And you know, when I speak, you're going to get tired. Now people are going to start logging off. Because we are reading as I speak, reading as I speak, reading as I speak. People in the comment sections of YouTube complain that the content is too much, but so far so good. You're the ones who chose the subject. The mere fact that you chose the subject, that means you accepted to take on all the trauma that comes along. But the good thing, I don't just put their words in howdy. We always make sure that each and every word we see on screen makes sense. So by that, if we do that for all topics, and we do that in 1 year, and you have a full year to revise, then how will you fail? How will you fail? You just now read and wait for God's plan. You see, we plan and God decides. So for you, you've planned your academics well, you just wait for God's decision. You can't really fail. So are we so far so good? Especially those people who went off are people who knew the topic. And we are talking of structures, which they even talked about first. So we are boring them. So I'm talking to those people who have been hearing eukaryotes for the very first time. Are we together? Should we really proceed? Are we still together? Are we on the same page? Are you still thinking and breathing? Okay, someone was like, I give them some 2 minutes to rest, and I think the 2 minutes are done. Yeah, the screenshots we shall take at the end of the class. I think that is okay, and better we shall take the screenshots at the end of the class. Don't worry. Yeah, don't worry. Don't worry about that. Okay. Now, Unlimited, you should be wise enough. Every time you get a chance, you take a screenshot. By the time the class is done, you don't stay to wait for the screenshots. Because for you be sorted. For you be sorted. So you don't really have to wait for the screenshots. You be sorted, for you just proceed. Okay. Now the class has just begun. We have just begun. Katandi kabutandis. That's what I think they say in vernacular. Okay. They are saying that these are the distinguishing features, or the features that really distinguish eukaryotic cells from other cells. Are we together? And they are saying, for them, they have it in mind. They have membrane-bound organelles. That's what I told you in the very first place. But they are saying the membrane is either one, what they call single membrane bound, or the membrane is a double one, like the nuclear envelope. Yeah? And they are saying those organelles that have a single membrane, only one, we have the ER, okay? Then we have the Golgi body. The membrane, if you were very clear, it was only one. Yes, there were cavities, but the membrane surrounding the cavities, it was only a single membrane. But the mitochondria and the core, plus this one, we have oversunk. We have oversunk that. It is a double membrane where one is folded, the inner one. Is it okay? Then we have said, do you remember when I told you that the prokaryotes have DNA that is naked? I taught you in the lesson of prokaryotes. And I said why we say that it is naked, it is simply because it is not having proteins they call histones. But when you reach eukaryotes, for them their DNA is not naked. But let's appreciate first and foremost that it is linear. The other no prokaryotes is circular. Are we together? I'm going to be explaining as I talk about prokaryotes for some cases. So in the end, it will be you people to make sense out of the comparison. Because comparison of eukaryotes and prokaryotes is very key in this topic. Very key. So they are saying for this one, it's not naked because it's having histone proteins. And we are also having RNA present. I told you about RNA and DNA in the previous class. I will not repeat that. I told you that each and everything I get a chance to explain for the first time, I give it my time so that the next time I meet it, I instead refer you and don't repeat myself. Okay? So the chromosomes that we have been seeing as chromatin, divided into euchromatin and heterochromatin, they are enclosed within the nuclear envelope. All of you know that, and you all know it is a double what? Membrane. You saw that prokaryotes had ribosomes, and we said they were smaller, and we said their size was 70S. But we are saying for the eukaryotes, the ribosomes, for them, they are kind of what? Larger. And the size is 80 what? 80S. Then we said prokaryotes, for them, they had peptidoglycan. But for eukaryotes, if the cell wall is present, why are we saying if present? Because animal cells lack it, but the plant cells have it. Are we together, people? So if the cell wall is present, it's having cellulose. And it is cellulose for the plant cells or the protist cells. But protist is ancient. The modern is protoctist. So for the plant or protoctist cells, they have the cell wall, but it doesn't have peptidoglycan or murine like the prokaryotes, but instead we have the what? The cellulose. I've told you now content has started, you listen. And this is what I beg. If you can maintain about 20 less than the number that you have now, 20 less, it is really good, and I'll be very happy. What I mean that by the time we end, if 20 people have left, I'm very okay. But if more than 20 leave, it is really bad. Because now I'm starting to speak. And I've told you, be wise. When I expose something like this, you take a screenshot so that I explain when you already have it. Later on, as you make your own notes, you also add in that extra information that I added. And it is always better you do it immediately after class so that you can remember some of the statements that I said. If you really forget them, you rewatch the video. But at a speed of time was 2. That means if the lesson was 4 hours, for you, you're going to use 2 hours. But you're just rewatching to add on those extra things you didn't really note down. Because they are really high yield and they will be helping you in all the content. Are we together? Then for the most fungal cells, they cannot have the cellulose. For them, they have what they call chitin. Hope we are together. Yeah. So those who really want to become agriculturalists, those who want to become botanists, you can now go an extra mile. Look at the structure of cellulose. Look at the structure of chitin. Things like that. Okay. We go. What are they saying? Oh, these things we finished. So this is what we are now explaining. So you can decide to take a screenshot now or take a screenshot later. And I'm not giving you the chance to take screenshots at the end. We are taking them right now. I've given you the first chance. This is the second chance. So this is what we are explaining. So let us read. What are they saying? They are saying that eukaryotic cells are found in majority of living organisms, and that one we are all aware. It's us, the eukaryotes, that are so common than even the prokaryotes. How many times do you see bacteria? But how many times do you see things belonging to Kingdom and Mary? How many times do you see people? How many times do you see dogs and other animals? All those are eukaryotes because they have eukaryotic cells, okay? So, of course, majority thing is just okay. Now, they are saying it is said that they evolved from prokaryotic cells. And this shouldn't confuse you if at all you attended the first class where I told you about the cell theory or the modern cell theory, where we say that all cells or all existing cells or all cells are derived from pre-existing ones. So if at all the prokaryotes or the prokaryotic cells were the simplest, most primitive and earliest, that means these other cells, chances are high that they evolved or came from them. That's why in one of the tasks you're taking home today is you going to make research about the endosymbiote theory so that you really appreciate about how the eukaryotic cells evolved from the prokaryotic ones. It is a very nice theory. It is called the endosymbiote theory. Okay, you will see it at the end of the class. Now, they are saying and telling you that the development of eukaryotic cells from prokaryotes came with a lot of things. And the very first one was development of membrane-bound organelles. If you recall, we say that prokaryotes lack the membrane what? Bound. Then we have said that this side, they are membrane-bound. Yeah. Are we together? And we have said we have those that are, okay, we talked about that, the single and double. I don't want to repeat that. Then the other development, it was development of the nuclear membrane. Remember the DNA in prokaryotes, for it was just there loitering around in the cytoplasm. But this one of eukaryotes, it got an envelope around it. So that was also a development that came from eukaryotes trying to evolve from what? The prokaryotes. Are we together? They are adding on and they are telling us that these chloroplasts or the chloroplasts and mitochondria, they are so special that they are semi-autonomous. Semi-autonomous. Semi-autonomous is something that is very good. You all know when they say semi, they mean something like half or part. Have I lied? Okay. When they say semi-autonomous, when they say semi, you think of something like half. You think of something like maybe partially, isn't it? Then when you see autonomous, you think of something like automatic. Are we together? You think of something like automatic. So partly automatic. So it's like they are telling you the chloroplast and mitochondria, they are partly automatic. If they tell you that something is automatic, I know that people here drive, but let me not use that example maybe. But something automatic is something that maybe that it can take place on its own. They tell you that computers are automatic simply because there are tasks they can do on their own. They can update the software on their own by simply you clicking update. You've heard of things like automatic updates. When you turn on automatic updates on your phone, you just wake up and it updated itself. You realize? So things happened without your consent. So these chloroplasts, these mitochondria, They are semi-autonomous. They are partly automatic. They can really do their own things without involving the cell, simply because they have their own control center. They have their own DNA. They have their own ribosomes. If they have their own DNA, that means they can do their own DNA replication. They can control their own cellular activities. Okay, they can control their own activities happening inside them. Sorry, non-cellular. If at all they have their own ribosomes, that means they can synthesize their own proteins. Remember, ribosomes are sites of protein what? Synthesis. But we are saying they are semi, not fully autonomous. So they do that partly. That means time will reach and they will need the cell. Yes, they are somewhat autonomous, but at some point they will need the cell. I don't know whether you people are trying to get, but we get shocked. We are saying that what is really giving them the guts and aura of being automatic or autonomous on their own is because of their own DNA they possess and because of their own what? Ribosomes. So because of the DNA, that means they can control activities occurring within them, and because of ribosomes, they can synthesize their own proteins. But do you know what shocks us? Remember, they are part of the eukaryotes. But then eukaryotes, we know that the eukaryotes have linear DNA. But we find out that inside the mitochondria and chloroplast, the DNA that is there is circular. It is circular and not linear. This is a red flag. Remember, the circular DNA belongs to prokaryotes. Then to make matters worse, the ribosomes inside the prokaryotes, the chloroplast and mitochondria, we expect them to be 80S because the 80S are the ones of eukaryotes, but we find them as 70S. But remember, 70S was for prokaryotes. So that is clear evidence that chances are high that eukaryotic cells came from prokaryotes, and we'll appreciate that more when you do my research. Which research? Go and find out what is the endosymbiote theory about. But I've given you the hint already. Yeah. So you know that these two belong to prokaryotes. Remember, they are inside chloroplasts and mitochondria, which are organelles for eukaryotes. Do you see some sort of clear information that maybe eukaryotes came from these guys? That is very key, and please take note of what? Take note of it. Let us see what they are saying. They are saying that they are semi-autonomous. Why? Because they can reproduce on their own. Why can they really reproduce on their own? One, they have their own DNA. Though it is circular, they have their own ribosomes, which are 70 what? 70S. That means they can make their own proteins, like enzymes. Don't you know that enzymes are protein in nature? Very cheap things. So we are having 4 forms of eukaryotic cells that you really need to learn. I told you eukaryotic cells are inside eukaryotes. So plants are eukaryotes, animals are eukaryotes. But inside you, the animal, you have blood, and your blood has red blood cells. So cells for that animal, the animal cells, are the eukaryotic cells. You have what we call the nervous system that includes your brain and spinal cord. And we have cells, they are called the neurons or the nerve cells. I taught that in coordination. You have the skeletal system with, of course, skeletal muscles. You know you have biceps, you have triceps, all those are muscles. You have the extensors, you have the flexors. You understand? And there are cells there. Then as a plant, the plant has so many parts. The plant has roots. Those roots have root hairs, and the root hairs have root hair what? Cells. The leaves have the stoma. The stoma is surrounded by guard cells. They are here. Leaves have the palisade and spongy mesophyll cell layers. And of course, we have mesophyll cells there. And we shall see that more when we reach nutrition. But I believe, you people, I don't know whether you're taking this app very serious. I told you about the Pinterest app where we get our images. You'll even be surprised that even the images in this book, I get them from that side and just modify them in one way or the other. So if at all you have issues with the mesophyll cell, root hair cell, you can put in root hair cell, look at it and proceed with the reading. Okay? Fungal cells. The most common fungi known by students is yeast. So we have the yeast cells. You know that fungi are good at producing spores, so we can have the spore producing cell. It's supposed to be spore producing. Eh? Then we have the hypho. What was wrong with me when I was writing this? It is hypho cell. Sorry. Why did I write that? I normally proofread my work, but it was some good time back. So we have the protist. This is ancient, and modernly it is the protoctist cells. We have the amoeba. I think you know that. We have the paramecia, and I know you know that. Singular for paramecia, it's paramecium. Hope you know that. We have the euglena or euglena, whatsoever you want to call it. So those are the forms of eukaryotic cells that we have. So many people in the country think amoeba is prokaryote, but I want to show you that amoeba is a single-celled eukaryote. Hope you distinguish single-celled from multi-celled, and you distinguish prokaryote from eukaryote. Okay, we go. What are they saying now? This is the interesting thing I wanted to tell you about. These are things that really make science so good. So you take a screenshot, then we explain, okay? What are they saying? What are they saying? They are saying the living contents of eukaryotic cells are divided into two. That is the nucleus and the cytoplasm. I want you to listen carefully. The living contents of the eukaryotic cells, there are only two. Nucleus and cytoplasm. Are we together? And together they form what they call the protoplasm. So when you hear protoplasm, have it in mind they are meaning living content of the cell, which is strictly the nucleus and cytoplasm. You people, they have not included the cell membrane, and they cannot include the cell wall because the cell wall is specific. For it decided to only be it decided to only be present in the plant cell and be absent in the animal cell. So we are not going to involve it in our things. Are we together? So when they say protoplasm, when you see the word plasm means the content. When you see plasm in science, it means content. You will find so many things to do with plasm. Neoplasm, that is at medical school. But at your level, you find things like nucleoplasm, you find things like azoplasm, axoplasm, things like that. By the way, that liquid that is found inside the nucleus, it's called the nucleoplasm. Be aware. Where the DNA, where the nucleolus are suspended, it's called nucleoplasm. So I've been telling you protoplasm. So protoplasm is the living content, and the living content is having only two things, that is the nucleus and cytoplasm. Then they're adding on the most interesting part, which I taught you already, that the soluble, or what they call the fluid part of the cytoplasm, is what they call the ground substance, or you can call it the cytosol, Fa, or rarely called the matrix. And what is the role of that fluid part or the soluble part? It is normally a store for vital chemicals like enzymes. That's why it is a site for so many metabolic reactions like glycolysis. Why I attached glycolysis here? Because it confuses so many people. They don't know where glycolysis really takes place. But you've learned that it takes place in the cytosol. It takes place in the ground substance, which is the fluid part of the cytoplasm. That's why some books just tell you that glycolysis takes place in the cytoplasm. Don't be so surprised and don't get mad because I'm saying glycolysis and you've not heard of it, but you will find it in respiration. I told you this is a topic that collects content from so many topics but brings them into here. But we have no option, yeah? So we shall always do that. Are we together? Now, when you see something, when you see plast, think of structure. So most people think protoplasm is the same as protoplast. You people, if you think biology is easy, you get anyone and tell that person what is the difference between protoplasm and protoplast. They will tell you it is not examinable. But you need to learn. These are things that really make you a super student of the subject. You're not a gambler. So plast and plasm, they are different. Plast means structure. How I used to recall this as a student, I used to do this. I used to think this plast structure. structure, then plasm content. So the moment I get the protoplasm, I am aware protoplasm is nucleus and cytoplasm. The moment I add there the cell membrane, but I subtract the cell wall, so I mean without the cell wall. I told you we are not involving the cell wall in this, because for it is, how do they call these people? Okay, it is a racist. It chose the plant cell, it left the animal cell. So we are also not going to involve it in our things. So when I get the protoplasm, that is the living content, the nucleus and cytoplasm, and I add on only and only the cell membrane, I get what they call the protoplast. Without the cell wall, let me hide this. Let me hide this. I need someone who is going to tell me what I've said. I need someone who can raise up their hand and you tell me about protoplasm and protoplast. I give you some money. Yes, that one is not going to be free because I've just said it. So someone who can really grasp it immediately deserves something small. Who can tell me protoplasm? And I think it was Shalom first. Shalom, unmute. Shalom, unmute. And then speak.
Yeah. Protoplast means structure and protoplasm means content.
So what is protoplasm? What is it composed of? Now those people who had taken the screenshot, they have gone back to gallery, they have copied. You see, Shalom has Shalom has disappeared. That's what I be telling you, that when I explain, you've not understood, but you've appreciated. So you're supposed to go back and capture the content. Are we together? Sorry, take that serious. Okay, let me hear from Fad. I want someone to say it the same way I've been saying it.
You say that
Mm.
protoplasm is the content, then protoplast is the structure. Meaning, when we
But you
find
people, you first listen. I didn't say protoplasm is content, and I didn't say protoplast is structure. But I said when you see something like plast, you think of a structure. When you say chloroplast, it is an organelle with its particular structure. When you see plasm, you think of content. Okay? Then protoplasm is the living content of the cell. And what does it have? That's what I want you to tell me.
the living content of the cell contains the nucleus and the cytoplasm.
And the cytoplasm, So what of the protoplast? What is it composed of and what doesn't it include?
The protoplast contains it doesn't include the cell wall.
Yes, but it includes what and what?
The cell membrane and the nucleus.
Only? Cell membrane and nucleus only? You see, you have forgotten.
I've forgotten it.
And the cytoplasm, it includes the cell membrane and the living content, which is the protoplasm. That is very cheap. You see, you people, I really wanted to give someone money, but you people are not allowing me to give you money. So you now listen, because a lot of content is coming down. You're supposed to listen and win some money. So protoplasm, which is the living part, plus the cell membrane without the cell, that is poroplast. So structure, just remember that we are trying to involve the cell membrane to have a complete structure. But without the enclosing membrane and it's just the living content, it is plasm, protoplasm. I think that is very clear. When you read, you will understand. Then the cytoplasm, this one I already told you, that the cytoplasm has the living cell organelles, it has the non-living inclusions, and it's having the soluble or fluidy part, the cytosol, isn't it? We said the cell organelles are living, but they are non-fluid. The cytoplasmic inclusions, they are non-living, but also non-fluid. Are we together? And we gave examples: glycogen granules, I told you why they are called like that. Starch granules, I told you why they are called like that. Lipid droplets, I told you what they are called like that. Cell organelles, I just put the mitochondrion, but there are so many. But I think this one is just obvious because we talked about it already. Yeah, protoplasm and protoplast. Hey, make sure you're sober. Hey, someone can just have the equation and says protoplast, because it's supposed to be a full structure. So someone tells you, to get the full structure, I will have to get my living content, which is the protoplasm, and enclose it. And I'm enclosing it with the cell what? Cell membrane. Why not cell wall? It is specific that it doesn't favor the animal cell. Hope we are together there. Very cheap. We go. So yeah, they are talking about the differences between the plant and animal cell. And this is one of the things they really like so much to talk about here. And I'm going to explain. So let's agree. Let me explain the plant and animal. For you will explain, or you will read more about prokaryote and eukaryote. Okay? What are they saying? The plant cell has a cell wall. This one doesn't have a cell wall. But remember, the plant cell wall has cellulose. So if this one doesn't have actually just say has cellulose cell wall, lacks cellulose cell wall, like that. Even if you don't split, lacks cellulose cell wall, has cellulose cell wall. Are we together? They're saying that the plant cell has plasmodesmata. Who knows plasmodesmata? Do I have anyone who knows it? Okay, that is good. We're having some people who know it, and that is very nice. So you know that we are having the hexagon like that, 6 sides, and we said that these plant cells had a cell wall, what, like that. So I'm assuming that that is my plant cell, and I'm also, let's say we are having another plant cell here. Maybe they are in touch. Okay. Oh, this one is looking like naphthalene. Okay, now this is what they call the plasmodesmata. Are you seeing that thing? This is plant cell 1. Are we together? And this is plant cell 2. So what is connecting them is what they call the plasmodesmata. You tell me in the chat, is plasmodesmata singular or plural? Let me see if really you people are doctors or botanists. Let me see if you're really botanists. That's what they call the plasmodesmata. Is plasmodesmata singular or plural? Eh? You people, you're not serious. Everyone is saying singular. Now let me ask. Let me show you that you guys cram. You tell me, is stomata plural or singular? Stomata. Because you may think that stomata is different from plasmodesmata in terms of biological English. Now, how can you tell me that stomata is plural, then you tell me plasmodesmata is singular? Does it really make sense? It doesn't. You people, plasmodesmata is plural. I did it intentionally so that you people can fall for the trap, and you've fallen for it. So if I'm naming one, I'm supposed to say plasmodesma. Plasmodesma. Yeah, like that. It is one. So it is connecting two plant cells, and it is connecting them via the cell walls. So I want you to be these kinds of students who will scroll at the end of this table. I remember putting the food. I have a very good memory. I put it here and I told students, plasmodesmata. You see? Is communicating that is a plural word. They are tiny and cytoplasmic. They are connecting adjacent. So the plant cells are supposed to be adjacent next to each other. And the connection is through what? Cell walls. So the mere fact that it is through cell walls, that means we cannot find plasmodesmata in animal cells. Nowhere will the cell wall be found. Nowhere. Okay? Let me hope we are together. In case I leave you, you raise up your hand and ask. I promise I will respond. Okay? So even this one has been explained. You will know that the plant cell has that one large vacuole that we call the cell vacuole. But the animal cell, it's also having its vacuole, but it is not the sap vacuole. You should note that. For it is just a vacuole and it is smaller. And for it is at the periphery, but this one is at the center. Okay? We are saying that the plant cell has what we call plastids, such as chloroplasts. Who knows plastids? If you know plastids, raise up your hand. I give you 500,000. If you know plastids, I am giving you 500,000. No one knows it. Even those who will be listening to the recorded class. You see, no one is raising up their hand. But let's learn it today. Plastids. You're seeing something to do with plast. And I warned you what plast means. When you see plast, think about structures. So plasts are things that are involved with manufacture in plants. That's why you can look for any organelle. You can look for an organelle in animals that has the word plast, and you may even fail to get it. But when they say plastids, strictly plastids, they have it in mind. They are just to do with food manufacture. And what did I write here? Those days when I was still a good writer. I said that plastids are membrane-bound organelles. They're just covered with a membrane, so they are organelles. Appreciate that. They are found in plant cells. That is very key. What are they really important for? They're important for food production. They're important for storage. Those are the plastids. Biology has a lot of things. Now someone is like, but as if we talked of plastids in the lesson of prokaryotes. Those were plasmids. The small rings of DNA that have that self-replicating property, if you recall. These are now plastids. They're in plant cells, they're in membrane bound, and they are responsible for production and storage of what? Food. The very first example that is common to you all are the chloroplasts. Who doesn't know they manufacture starch, which is food for the plant? We have what we call chromoplasts. I want you people to go to Google right now. Ask it what is the meaning of. Okay, don't go to Google. Don't go to Google. Have you heard of what they call chromatography? Have you heard of chromatography? Okay, what is to do with chromo? When you see chromo, what do you think? Or when they tell you about chromatography, what comes to your mind? What comes to your mind when you see chromatography, anything like that? What really comes to your mind? You tell me in the chat at color. Thank you, Davi. When you see chromo, you think of color. Biology is not hard at all. When you see chromo, you think of color. Even if you ask Google right now, they will tell you chromo, color. So the chromoplast, what do? Most of you have been there, and I know you have never asked yourself this question. That why do these tomatoes eventually turn red? Why do the apples eventually turn to that color? Why do carrots eventually turn orange? Things like that. So inside, those are plants. So inside them, they have plant cells. And their plant cells have what we call chromoplasts. So it's those chromoplasts that give them color. If you think biology is easy, tomorrow you ask a friend, you ask them, do you know why the tomato is red? They cannot mention chromoplast anyway. They will just tell you that's how God made them. But little do they know that God made chromoplasts and left the rest for the chromoplast. Are we together? Then we have what they call leucoplasts. Leuco. When you see leuco, it is so common. Leuco means white. It means pale. Anything pale, anything white, something like that. Pale, white, colorless, that's what leuco means. That's why white blood cells are called leukocytes. And red blood cells are called erythrocytes, because erythro means red. Everywhere that you go. That's why if you are told you're having a complication and maybe your palms become red, most of you have realized that, especially the light-skinned people. And when you go to that and we are trying to spot diagnosis at the hospital, one of the things we attach to your report is palmar erythema. You understand? Palmar erythema, trying to show that your palms went red. So when you see leuco is to do with white, to do with pale colorless. So these ones are now colorless. Yeah. And these ones are just good at storage. And somewhere there in the future, we shall see that there are some kinds of leucoplasts they call amyloplasts, but we are going to see what they do in the future. I don't want to say all the content here, yet we are going to meet it. People may hate the subject. So do you people see that in just 5 minutes, you've learned a lot of things? Plasmodies matter. You've learned that it is not even singular, it is plural. You've had plastids. You've had they are different from plasmids, and you've recalled plasmids were in prokaryotes. You've learned that they were smaller and they were ringular or annular, and they had a self-replicating property. You've now heard of plastids. You've heard their structures. They're only in plant cells. And you've learned that how to recall maybe plastids, plant cell, plastids, plant cell. You now know they are to do with food, producing it and storing it. You've heard that they are commonly three: the chloro, chromo, and the leuco. Chloroplasts, you know them, chromo color, so they give fruits color and flowers color. Why do you think you find pink flowers? It's because of chromoplasts. Then we have said the leuco, they are colorless. They are to do with storage. And an example is amyloplasts. And we shall see why what amylo means. Okay? Very good content. So we go back. Okay, Fahad, you have something to say. You say as I scroll back to our table. Yes, Fahad. So the people who will be listening to this in the future are supposed to be pausing. You'll be like, what has he said, but he didn't write? You note it. So, I think this one was a very big mistake, but the plant cell is the one that is regular shaped, and the animal cell is irregular shaped. Plant cell has the middle lamellae because we saw it is above of the cell wall. This one lacks it. We saw that the tonoplast is the one that surrounds the cell sap, so it's only in the plant and it's not there in the animal. We saw that animals, their nucleus is central because we don't have that large vacuum that pushes it to the periphery, but in the plant cell it is at the periphery. Our plant cells really make starch or carbohydrates. Of course, you know that starch is a polysaccharide, and polysaccharides are carbohydrates. And plants store them in form of starch, but these animals store them in form of glycogen. Or someone can say that here we have glycogen granules, but this side we have starch granules. Okay. What else? We talked of the centrioles. Centrioles are these ones, the ones we say that they are together, occurring in pairs and at right angles. So have it in mind that they are only present where? In animals, but they are absent in plants. Remember, for them, they play a role in cell division. So we shall talk more about centrioles when we reach cell division. So in your summary, you should attach it that when you're reading and you see centrioles and you don't kind of get them, you recall that doctor said that shall be taught in cell division. And cell division, me, I completed it. I completed cell division long time. So you just need to have access to the drive and watch it. Yes, let me see. I think cell division we had 3 modules. Let me confirm it. Because now people may ask me for the work later and I've forgotten. But I remember covering. I covered. So when I check, where is biology? I can't see biology. It's here. It is senior 6 work, and I think it is term 2. Yeah, inheritance and evolution. Yeah, I covered cell division. Yeah, there are 3 modules. All those are classes ready. So it would even be very good that after this lesson of eukaryotes, you find ways of accessing the drive. You just talk to me. I give you the drive, you also capture cell division. By the time your teacher thinks of introducing it for you, you're knowledgeable. You just sit in class. You just doze. But your friends don't know where the marks are coming from. But please, you also involve your friends and tell them about SSA. Yeah, don't just be too bad and let your friends fail. Now we are saying that this side we are having, okay, later on we shall have cilia and flagella for the higher animals, but in higher plants the cilia and flagella shall be lacking. Now that is just a fact, because someone may ask me like why, but that is a fact. because our research was clear that higher plants, Nocidia and Flagella, but higher animals, the cells had those things. Yes, wisdom. So you all know that animal cells will have fewer organelles, but the plant cells will have more ones. Why? Plants have cell wall, the other ones don't have. Plants have tonoplasts, the other ones don't have. Plants have chloroplasts, the other ones don't have. You realize so many things. Okay, the plant cell has so many things that animal cells don't have. So you realize that the other ones will have more. The same with eukaryotes and prokaryotes. Eukaryotes will have more organelles. Please, you talk wisdom. You're already unmuted.
Yes, master.
Don't call me master. I don't teach karate. I teach science
Okay.
and math. Call me tutor or
Okay.
doctor. Don't call me master again. I don't. Have I ever taught you kung fu?
Okay. Sorry, please.
So
Yeah,
don't
for me,
call me master.
my inquiry was I wanted just to know more like is it like you talked about plasmodesmata. Is it the same as the chroma? Is it the same as this pigment which helps in photosynthesis?
It is not.
Okay, sir.
The pigment is chlorophyll, yeah?
Not chlorophyll,
But
it is chlorophyll.
it is what?
It is something like porous matter, something like that.
Oh, which textbook? I
Okay, it
try to
is.
check my memory.
I just got it on some app.
Oh, it is okay. But I've not seen that in any textbooks, but it can be existing.
Okay.
It can be existing. So,
Maybe I can pronounce it once more. Is chromo? It is chromoplasts.
it's the one I gave. Didn't you see the
Okay.
chromoplasts? It is here.
Yeah. That's why I'm like comparing it. Is it the same as the plasmodesmata?
Plasmodesmata is different. For it is just connecting adjacent plant cells through there. It is connecting adjacent plant cells through their cell walls. The chromoplast is a plastid that gives color to fruits and flowers. It is also inside plant cells. They are organelles and they are membrane bound. It is clear there. I think you
So
kind of dozed off. Hope it is okay now. I don't know why you've muted, but hope it is okay now. I think we can proceed. So the definition of the organelle, I've already said this, and you know why it is distinct. You know why the structure is particular. You know why the function is particular. And they are telling you so many organelles for the eukaryotes are membrane what? Bound. And people know that. So we are now going for the functions. So this is where the lesson begins from. Functions. This is now where you're going to find content. Because now, trust me, the table is very long. That's what I can recall. Oh, that is a table. Oh, these are functions. This is the third page. This is the fourth page. Functions. This is the fifth page. Functions. Sixth page. Functions. Seventh page. Functions. Eighth page. Functions. And finally, ninth page. Function. So you have 10 pages almost to listen to work, and you're supposed to read. Then the comparison between prokaryotes and eukaryotes, I want you to go and just develop that on your own. And you see, when the comparison is done, the topic is done. Yes. You see, after all that, the task is there for endosymbiote theory, and the next thing shall be this cell surface membrane, where we shall talk about the fluid mosaic model. So you're having 10 pages. I told you I'm not going to leave this place unless all that work is explained. I think you guys are not aware of my true colors. But trust me, these things we are going to just be talking. All of them, I've said them. In Luganda, they say chesta yogedenga tekiriyo. Yeah. So we are going to just be enjoying. There are 10 pages, but they are going to take 10 minutes. A minute per page. So in 10 minutes, the class is ending. Okay? And people are going, I don't know why. Maybe people are running out of data. Hey, tomorrow is a Monday, people have school. It is okay, but stay on, you endure. Ivan, you had something to ask? You raise up your hand again if you still have the question. or we should proceed? Yes, Ivan. So as we have a break of 30 seconds, let's hear from Ivan. Yes, Ivan? But please tell your
Yes.
friends to come back and study. Yes.
Good evening, tutor. The question I have is about the chromoplast. For me, the research I made before this lesson, I found out that virtues contain anthocyanin, which also helps in coloring of the fruits, petals.
Mm.
Now
Mm.
I you to clarify more about the
Eh,
anthocyanin in virtues and the
okay.
chromoplast.
I will talk about anthocyanin in coordination in plants, where we shall talk about flowering. Is it okay?
Okay.
Because because that is now a chemical, and in coordination, that's where we talk about those different chemicals: the anthocyanin, the ethylene, the IAA, that is indole acetic acid, the ABA, the abscisic acid, and so many others. So we shall also talk about that anthocyanin. That said, in flowering, you see how the color of flowers comes about, and hence those other colors you're meaning. But as of now, we may not need that. Okay? You're gone. You people, when you ask, you first stay on before you mute yourself. We first agree. Because you may mute yourself and then you start cursing me. And I'm not really sure whether we have really agreed. So please tell your friends to come. Most of you, you're in debt, but you've never told your friends about these classes. You just want to benefit alone. Please involve your friends. Yeah? There are a lot of things that come with these academics. The most important thing are the people we study with. We may all pass, but they get a better job. And the better job they get, they need, there is some vacancy. Then they tell you about the job. So involve your friends at an early stage. If you all pass, there is no problem. But if you pass alone, that is a problem. They are going to hate you. So involve your friends. Let's all benefit as a country. Okay? It is so bad for you to get access to a free class and you don't involve a friend. You know, you're on YouTube watching very good classes and you don't tell a friend about the classes. That is extremely bad. Okay? So be good people. And for me, I know those people who keep on referring friends. When you graduate, I also give you connections. Because even after Form 6, we are supposed to stay friends. Because you never know, there are some other classes I do even beyond high school. Okay? So be good people. Now, cell wall. Now, these functions, the curriculum asked us to really teach you functions of different cell walls in different areas. So even in topics that are not part of the syllabus. So some of the things I'm going to be mentioning, I'll be telling you to wait for a certain topic so that you don't just cram. Where I can say something small, I'm going to say it. Are we together? But let me tell you, these are things you just need to go and read on your own, but they are very clear and clean. Okay? Let us go. Said wall. What are they saying? Mechanical strength. Don't you know that? It provides skeletal support. Someone may be like, I thought skeletons are in people. Why are you talking about a skeleton in a cell wall that it is in plants? If a skeleton is simply a framework that is supposed to give you support, period. Okay, so the cell wall gives support to individual cells and the whole plant, the whole shrub, the whole tree. Remember, I told you there's a possibility for these plant cells to be together. So when they are linked up together, the cell walls are connected to form like one elongated cellular wall that can give support to the whole plant, to the whole shrub, to the whole tree. I'm done. It allows turbidity. I told you about turbidity in chemicals of life underwater. That when a plant cell takes in water, I want you to look at this. This is the plant cell. So look at this at the cell wall. Look at this at the plasma membrane. So the moment it takes in water, you know what is going to happen? This is what is going to happen. I really hate it to have things that are disorganized. So let me write it well. Water like that. So when it takes up water, you know what is going to happen? Remember, the cell wall is rigid. Remember, it is having cellulose, very rigid. For it will not move. But the plasma membrane may move to one extent of even wanting to touch the cell what? The cell wall. Now, this state is what they call turgidity. They say now that the cell is what is turgid. It takes in water, it swells. When it swells, it becomes turgid. Are we together? Okay. And okay, this is it. They're saying by resisting expansion. Remember, I've said for it is rigid, it doesn't move. But the plasma membrane inside, for it can what? Remember, for it is just there lazy. When water comes in, it can be easily expanded and the cell does what? Swells. But the cell wall, the one we are talking about, for it will resist expansion when the plant cell is taking up water. And you know that the support water provides the hydrostatic because of the word hydro. When you see hydro, just know what has been involved. And when that support is provided, the cell cannot burst. Even if you do what? With that cell wall that is rigid and has not decided to expand, the cell cannot burst, my dear. You see? Things are really making sense. We are still on the cell wall. What are they saying? They are saying the orientation of cellulose microfibrils limits and controls cell growth and shape. Someone will be like, now what is that? Microfibrils, they are talking of orientation. Orientation is arrangement. Look at this as the cellulose alone. Right now, you go to Pinterest and put in cellulose microfibrils. You look at them, you see how they look like. For me, I saw those things long time ago. But I was showing them to students those days. But now I want you to be looking for these things on your own. You see that if that one is maybe, let's say, cellulose molecule, and then we add another one, cellulose. Now this is what they call a group of cellulose microfibrils. Do you understand? So the way they are arranged, the way they are oriented, does us good or does the plant good by controlling cell growth and shape. You understand? Cellulose. Cellulose microfibrils. When there are too many, they can even become macrofibrils. I remember telling you that micro small, macro big. That was still under chemicals of life. Okay? They are still adding on. They are saying that those cell walls have spaces, and those spaces can help us to transport water. Transporting water, that means we are talking of transporting plants, but that is a topic that is not part of your curriculum. By the way, there are things you're going to miss, like these pathways, the apoplast and symplast pathways. They were really so good. The content was just nice, but you're going to miss them. But you will meet them at higher levels of education, you will understand. No problem. So those spaces in the cell wall can help us transport what? Some water, either by the apoplast pathway or the symplast pathway. So I shouldn't waste my time to tell you that, because it's not even part of your syllabus. Go to YouTube, put in animation of apoplast pathway, you see how the water moves. You see it, you appreciate, you leave it there. After all, you've seen how the water moves, so you have approved that water actually moves. If you don't recall the word apoplast and symplast, they cannot be marking points in your curriculum. You just say spaces for transportation of water, period. And I remember telling you in the lesson of water, chemicals of life, that why I keep referring you to those classes, they're on YouTube, so that when you realize that a point has been shallow, you're supposed to go and revisit and find out what did the tutor really mean here. Are we together? So xylem was for water and salt. Phloem was for manufactured food, like sucrose, things like that. So they are telling you that the walls of xylem vessels and those of the sieve tubes, but we shall see these ones in histology. Someone will be like, now what are those sieve tubes? Now the sieve tubes are also important for transportation. I remember telling you about this terminology of translocation still in that class. And we've just talked of transport here. So if they can transport water, what can really stop them from allowing translocation? Yeah? So it is just clear. I told you these first classes, make sure the points make sense. you're not supposed to recall now. You're supposed to recall after you've read. Is it okay? They are now talking of those of epidermal cells. Someone may be like, what are epidermal cells? You people, you have a skin. And you know that the skin is divided into the epidermis and the dermis. The dermis is down, okay? Then the epidermis is on top of the dermis. Is it okay? Epi in biology means above. That's why epidural germination, we are having the cotyledon coming where? Out. That's why in the heart we have what we call the cardiac muscles or the cardiac walls. And we have the epicardium, which is on top of all the others. Are we together? Now, when I say epidermal cells, they're just on top. Yeah? If you get a plant leaf, just on top of the plant leaf, the very first cells we expect to meet are the epidermal cells. So we are saying those plant cells just on top of the plant leaf, they have what we call a waxy cuticle. Waxy cuticle, very important. As you progress, you find out that it is made up of what they call chitin. In future, you'll see all that. But as of now, don't overmine chitin. So, what is the characteristic of this waxy cuticle? It is waterproof. Because it is waterproof, it will help us to reduce water loss from the plant leaf. But then, there was a student who asked me this in 2023, that if it is waterproof and it is preventing water loss, how will water enter the leaf? Who can raise up their hand and tell me, as you also rest a bit? If the cuticle is on top of the leaf, okay, the leaf on top, there are epidermal cells, and the other epidermal cells have a waxy cuticle, which is waterproof, so it prevents water from being lost from the leaf. Now, how will water enter the leaf if the cuticle is waterproof? Who can raise up there and who can think and tell us? How will water reach the leaf if at all the cuticle is waterproof? Yes, Achen. Yes, Achen. You unmute and speak. I've asked you to unmute. Yes, you speak. You're unmuted.
I was told that
Mm.
the waxy cuticle are part of, like they contain their lipids and they are phospholipids. So they have the
The
tail
hydrophilic
and
part, the phosphate group that can allow water.
yes, and the
Okay,
inside
good.
has long chains of hydrocarbons which can absorb the polar inside. Something
You
like
can
that.
reread. You can reread that part. That part of the hydrophobic interior is not correct, and that's why I wanted us to have it even as a special class. But you correct it. But okay, you've tried somehow. Let me hear from Mariam. Yes, Mariam. You unmute and give us an idea. If the cuticle is really waterproof and it is reducing water loss, where is the water going to come from? Yes.
So I think the water maybe will come from the roots via the xylem
Thank you so
vessel.
much. Thank you so much. She has said it. The water is coming from down from the soil. So it comes from the soil via the root hair cells like that. It comes and it goes up the plant. That's why transport is called transport up the plant. Yeah, thank you so much. Okay, then it is also a physical barrier. Because it is a physical barrier, it can trap things like bacteria, fungi, and that really reduces the risk of infection. Yeah? Because you've heard of bacterial infections, viral infections, fungal infections. So because it is a physical barrier, it can trap those pathogens, the bacteria, the fungi, the viruses. Is it okay? So here they are talking of Koch cells, they are talking of suberin. And I don't want to really traumatize you, but I want to give you good news that they are saying those things are talked about in secondary growth. So we shall see this in the 8th topic. The 8th topic is called growth in plants and development in insects. That's why we shall see primary growth and secondary growth. And trust me, we shall meet the Koch cells, we shall meet the suberin, but they have told you it serves a similar function, just like the waxy cuticle, so you can have something. Okay? The other ones were epidermal cells with waxy cuticle. Now these ones are Koch cells with suberin. But suberin and waxy cuticle have the same functions. Are we together? Yeah. So even here they are still talking over suberin, and we shall see suberin in detail. Don't really mind. But then these things would be really understood so much if transport in plants was still part. So I really even support you to leave out those points that don't belong to the topics that are outside. But the curriculum wants you to have it all. Because biology as a subject is supposed to also produce people who are going to go to the plant side. Not everyone is going to go to the clinical medicine or the clinical medicine. There are those who are supposed to go to herbal medicine. Herbal medicine is very good and it's one of the next big things in Africa. So you need this plant side. So I had to include this in our notes. Are we together? So they are what we call hemicelluloses, and they are just in cellulose of some seeds, not all. And the hemicelluloses are just food reserves. By reserves, we mean reservoirs. By reservoirs, we mean stores, things like that. Hemicelluloses, they are in cellulose of just some of them. Okay? Yes, Davi, what do you have to say as we proceed? Please, Davi, you had something to say? Unmute. I've asked you to unmute. Please speak. We have to proceed.
Yes, doctor. I want to ask what causes necrosis?
Oh, someone is asking what causes necrosis. Now we have what we call cell death. Cell death is divided into apoptosis and necrosis. Now, apoptosis is programmed cell death, and we are going to also see it here under lysosomes. Then necrosis, for it, it is uncontrolled cellular growth. We have uncontrolled growth of cells. You understand? That's why necrosis in the end it leads to cancer. Remember why people
it's
get
like
cancer?
a mutation.
Exactly, but the cells that are really growing uncontrollably have mutations. You understand?
Yes.
So they keep on growing and growing. So the tissue that is in place of being affected, in the end, it becomes cancerous. You get the point? Yeah.
Yes.
But apoptosis is programmed, so it has no effect to the body. But necrosis is so bad. It leaves it even damages the surrounding tissues. You understand? So necrosis leads to cancer. That's what I can say for now.
Then lastly, the cell wall. Does it have any role to play with the reducing necrosis?
Of course, according to what you've heard.
Yes.
I've told you necrosis, they are just mutated cells. Are you listening? And they are growing uncontrollably. They are going to grow too much to an extent of destroying even the surrounding tissue. So the surrounding tissue can be protected. Remember, a tissue is a group of cells. So the group of cells can protect themselves in that line. You understand? But have it in mind, in man, we don't have cell walls on our cells. So the cell walls, they are meaning are some other cells with those cell walls. So never talk of necrosis in man. I don't know that you're trying to get.
Yes.
You've read somewhere, and cell walls are helping us to combat necrosis. But remember, animal cells don't have cell walls. So you should think of it in other perspectives that don't relate to man.
Oh, okay.
Welcome. Now let us proceed. Now here they are talking of you people. That has been a by the way. Yeah, don't think it is part of what we are covering, but it is a good idea because it is about cell death, and we are studying cells. So I had to say something small about it. Okay. Now transfer cells. Transfer cells, these ones we shall also see them in transport. Yeah. We shall also see them in transport. So for now we may not say much about them. These notes of ours are textbooks with full content. You'll even find out that T cells or the transfer cells are somewhere. You can even just go to Pinterest, put in transfer cells, look at them. But it is all to do with transport. And transport, I've already mentioned it, that the cell wall is just good at it. Are we together? So let's go to the cell membrane. The cell wall is done. Cell surface membrane. We all know this one is selectively permeable. We shall even see this as we describe it in the fluid mosaic model. It is selectively what? Permeable. meaning it will let some materials in and not others. They can say selectively permeable, they can say partially permeable, they can say semi-permeable. So all those words, you please take them on. Selectively permeable, partially permeable, then also semi-permeable. Meaning the movement of materials in and out is restricted or regulated, and that is the main function. Okay, then another one is detection of stimuli. Now, in the next class under eukaryotes, it will be about the plasma membrane. You see that this plasma membrane has a lot of things on its, okay, it is on the surface. Some of you already know that it is a phospholipid bilayer. You know that. And you know it has some molecules on it, but we shall see them in the next class. But let me give you some of them, because I even mentioned them in the lesson of chemicals of life. But under research work, it has things like glycolipids. The plasma membrane, it has things like glycoproteins. And these things play a very important role. And you people know that these things in the end can even act as receptors, they can act as signalers, they play a lot of things, and we shall see them in the next class. Okay? Have it in mind that the cell membrane even has receptors. Receptors are just there to respond to stimulus. As we hear the word receptors, they are just there to receive. Are we together? So when we say detection of stimulus, it's because of those receptors that are on the cell surface membrane. At your level, you just need to know receptors. You don't need to go into deep details of which receptors exactly. Okay? They are telling you that membrane is good at transmission of nerve impulses, but this one will be taught under coordination. So we shall see all ways through which the cell membrane really participates. But if I'm to give you a simple hint, and I taught coordination in animals, and I completed. We talked about that transmission of nervous impulses. It is all about the diffusion of the sodium and potassium ions, only that. And it was taught very well. Let me show you here something small. I think it was, remember coordination is a topic of term 1. Eh? So if I come here, senior 6 term 1, it's their coordination in animals. You see, 3 classes. And they're already there waiting for you, and you've not watched any. And they are in order from introduction. Why don't you watch them? By the time your teacher begins them, you have all the ideas. You just enjoy the classes and you read work, you understand, you pass. So we shall meet it. Actually, you will meet it. We already met it. So it's you has not yet met us. Okay? Recognition of other cells. So I've told you that these things on the cell surface membrane, as we shall see the glycolipids, the glycoproteins, we shall see how they come about. Okay? They're the ones that play that role in recognition. So the membrane also allows cells to really attach to one another so they can form tissues. So someone can be like, how do they really attach to each other? They have what we call junctions. We shall see the tight and jump junctions in the same topic. They are the ones that really enable those cells to really attach to each other. Involved in energy transfer. Who doesn't really know that? If movement of materials in and out of the cell can really happen, what will really stop energy to be transferred across it? Involved in cell division. We shall see. I told you cell division was taught. So even this one, it is very clear. But if you want a simple hint of how it really participates, if this is the cell membrane and it is supposed to divide into 2, do you see that it really splits itself? It first becomes something like this. Oh, that is participating. Then it finally does what? Forms the 2. Don't you see it really participating? So it participates, so it's involved. It plays a role in immune responses. We shall see this in a topic called defense under transport in humans. And we shall see that in immune response. I don't know whether you people are aware of things like antibodies and antigens, but I believe you have issues with them. So in that topic, we shall talk about them. That's why I gave you a disclaimer at the beginning of this class that so many words are going to come here, but it is a topic that is talking about a cell. Which cell is in each and every topic? So some things we will need us to first reach that topic for them to make sense. Are we together? So we shall first understand what an antibody is, what an antigen is. Then we shall get everything. But all I know you people have heard of what they call have you people heard of what they call the T cells? T cells, have you heard of them? T cells, or at least the lymphocytes. I don't think you people have, but we shall see it in that topic. It is called immunity and defense, or defense and immunity, and it is under transport in humans, and it is part of your syllabus. Okay, it is a site for enzymes. It is a site for some enzyme controlled reactions. I think I'm starting to get tired. Yeah, but we are going to complete, trust me. So some enzyme controlled reactions take place there. Yeah, they take place there. Some may be like, like which ones? There are some kinds of reactions that we shall look at, especially under respiration. That's why they have said some. They are not so many. Someone may be like, why would we really have a process? Why would we really have a process that is taking place? Why would we really have a process that is taking place just in the membrane? It is very clear. We shall see it under respiration, yeah? Then you will appreciate. Just because I don't want to use so many terminologies. But we talked of glycolysis, which takes place in the cytoplasm. Now that glycolysis turns glucose into pyruvate. Now pyruvate is supposed to be handled in a certain way. And by the time it enters what we shall call the Krebs cycle, you will see some reactions in the membrane, and we'll appreciate that. It really helps us in such things, okay? They are talking of insulation of nerves, and I've told you when you see nerves, what do you think about? Coordination, nervous coordination, and it was already covered. But to give you some hint, Achen told us that she heard that the membrane has what they call a phospholipid bilayer. Phospholipid, that means lipids are there. And you all know that lipids are fats, and you know that fats are good at insulation. Do you see that? But we shall also see in coordination that we have what we call the myelin sheath. And we shall see that a myelin sheath is secreted by what they call the Schwann cells. And of course, Schwann cells have what they call a cell membrane because they are cells. Do you see that? So content will really connect when you reach those further topics, and then you come back to cytology, then you become a very good one. Are we together? Now we have the microvilli. Now microvilli, I told you, just have one thing in mind, that they are good at absorption. You understand? That even in your intestine, you go to Pinterest and ask image of microvilli on intestine. They will show you that our intestines have microvilli, and it is the microvilli that is supposed to help us with the absorption. They're even telling you that the microvilli is even there in kidney nephrons, not only the intestines I'm talking about. And it's not only absorption, but also what? Secretion. And I told you what secretion means. So as we are secreting something, the microvilli can help us. Secretion is out of the cell. Absorption is into the cell. So all can happen just via the microvilli. After all, one is the reverse of the other. And it's not only in intestines, but also in the kidney nephrons. These things of epithelial cells, we shall see this in histology. This is where we look at the epithelial cells and so many other cells, all kinds of tissues in histology. Okay, now they are also adding something so important. Just make sure they make sense. Later, you're supposed to read again to now master. You understand? They are saying that microvilli contain actin filaments. I remember telling you, I've not yet told you about the cytoskeleton, but we are meeting it very soon. When you see this, why the IN is bold? Every time you see IN in biology, think of what they call a protein. Because even when you look at the protein, it ends with IN. Most of the proteins end in IN. Look at insulin. Look at albumin. All those are in your curriculum. Look at globulin. It transports most of the things. It ends with IN. Look at keratin, the one in your hair. It ends with IN. Look at tubulin. Look at actin. Look at myosin. Very many proteins. Any protein that you know. Most of them end in IN, just like the word protein. So when you see actin in protein, are we together? we shall see, just because your curriculum doesn't have the topic that was called locomotion and what and support. You will forgive me for always saying the curriculum doesn't have, because in the old curriculum we used to connect those topics. So as teachers, we are finding it very hard for us to do without certain topics. So we are supposed to be bringing them as parables. But there was a topic they called support and locomotion. And they used to talk about movement and locomotion in man, where we talk about contraction of muscles. As you're running, as you're writing, as you're doing what? Muscles are contracting. So in contraction, we had what we call the actin and myosin filaments. They used to interact, then contraction happens. Are you following? But don't really feel bad that the content is really getting too much. Be like, at least microvilli, I have appreciated secretion and absorption. This actin filament is just a, by the way, extraordinary content. But when you see actin, think of myosin, think of contraction. And when contraction happens, even movement can happen. Are we together? Mitochondrion, this one you're aware that some stages of aerobic respiration happen there. And they have specified the stages, that is the Krebs cycle and the electron transport chain. And we shall see that in the topic of respiration. As of now, just appreciate that it is a site of some of the stages of strictly the aerobic type of respiration. And we shall see why not the anaerobic type. But even if you leave this class, when for you've just captured one thing, I want you to listen. When for you've just captured only and only one thing, and that is mitochondrion being a site of respiration, that is also something good. But if you can go as far as some stages of the aerobic respiration, that is again better. Are we together? Now, I want to tell you about this lysosome. Very well. It's one of the things that you're supposed to leave this class when you're well conversant with. Lysosome. Okay? Lysosome. Now, I told you that for you don't focus about this some. It is almost on most of the organelles of the cell. I told you when you see that this word lysis, when you see it, just know they are talking of breakdown. Yeah. They are talking of breakdown. That one I told you. Breakdown. Who is going to break down? In most of the cases, they are enzymes. If it is hydrolysis, because of hydro, water is doing the lysis or the breakdown. So in this case, they are going to be enzymes, and these enzymes have a name. Because these enzymes are going to help us with lysis, in biology we call them lytic enzymes. Lytic. Very important, lytic enzymes. Because they are going to help us with what? With breakdown. Remember this lysosome, if you recall our structure, it was just like this. It was just a small circle. Just like that, that's how it was. And have it in mind that these lytic enzymes are inside there. That one you should know. They are inside there. So it is just there. Very dangerous. Very dangerous. It is having enzymes that can do breakdown. That's why, because these lytic enzymes that can break down are inside it, that's why they call it a suicide bag. Someone usually asks me, why are they really called suicide bags? They're called suicide bags because inside they have dangerous enzymes. Why are those enzymes dangerous? They are lytic. What does lytic mean? They can break down anything they really encounter. But this breakdown is different. And I want you to listen very carefully. So all the functions of lysosomes are going to be looking at lytic enzymes breaking down something. And that is all. Though this breakdown is going to be divided, there is what we call autophagy. Yeah. There is what they call autophagy. And then there is also what they are going to be calling autolysis. And you need these things, I'm telling you, but they are very cheap. Just listen to me and you get to know how we are deriving everything. So when you look at auto, remember they are inside the cell, yeah? So don't think about, okay, when we see automatic, things are just happening on their own. You understand that they may not need the cell to really initiate anything. For them, they can do anything on their own. So the breakdown that is going to happen, it is either autolysis or autophagy. Now, What is this autophagy? Now, autophagy is this. When you see phagy, okay, you first tell me, you go to Google and ask the meaning of the word phage in biology. Phage. I mean this phage alone. Yeah. Phage. You go anywhere and ask. You go and ask, what is the meaning of phage? Because people may think these things really come from, these things don't come from heaven. These are normal things. Normal English that we have been explaining every time. Do I have some answers in the chat? Oh, most people, their phones are blacking out. Sorry. Someone said pepsin. You see, pepsin is ending with IN. There is also trypsin. It's also ending in IN. Okay. You people, phage, what does it mean? You go and ask and then you come back here and tell me very fast. Phage. Just write it in the chat. I'll have a look at it. Phage. And I also tell you here that phage, I told you about phagocytosis, and I told you that phagocytosis means cell eating. And I told you that how does the cell eat? I told you that it first forms a pocket, then it seals off. So the pocket comes, and inside there is some food. Are you together? If you recall, some food. Okay, so this one is called the food vacuole. And I told you if it is cell drinking, because of drink, inside we shall not have food, we shall have a drink. So for that one is called a vesicle. But you can add on and call it pinocytic vesicle because the drinking is pinocytosis. And here the vacuole, you can add on and say phagocytic vacuole because the eating is phagocytosis. What is the meaning of phage? Someone has asked something else. Someone has asked bacteriophage, and maybe you're asking, yeah, and it's not so sure. But phage, it's always about eating. That's why when they tell you bacteriophage, it is a virus that eats bacteria. The bacteriophage virus. It is a virus that eats bacteria. You understand? Phage is always to do with eating. That's why someone in the chat found something that says a virus that infects bacteria, but you searched poorly. For me, I just wanted the word phage only. Someone is saying something that eats or consumes. Yeah. So that one is to do with food. So when they say autophagy, this is what they mean. They mean that if this is the lysosome, are we together? It's going to be like this. It will release the lytic enzymes. You understand? It will release the lytic enzymes. And then the lytic enzymes can break down that food. That is the thing. That's what I wanted to tell you. That after the cell has eaten, the lysosomes can play that very important role. They bring the lytic enzymes, then they break down the food into more soluble components that really the cell can utilize or assimilate. Are we together? Then autolysis. Hey, when you see that, remember lysosomes, we already are aware they are good at breaking down. And again, we are seeing one of the functions as being autolysis. So when you see autolysis, you recall that doctor told us that with autolysis or autolysis, they are breaking themselves down. That is it, nothing much. They are breaking down themselves. That's why they call them suicide bugs. So do you see how bad lysosomes are? That they can release lytic enzymes to break down something else outside them. For example, if food inside a food vacuole. But again, they can also do what they call autolysis. They break down themselves, which is very bad. And we are going to see this one in so many scenarios still in our notes. And we are going to appreciate everything. Okay? If I leave you behind, you say something. Then I see how to help. So what are they saying? They are saying that lysosomes are suicide bugs. And everyone has appreciated that. You know why? So they are saying that they digest food which the phagocytic cells like the amoeba consumes from the surrounding. You get? The amoeba will consume food from the surrounding. After, since, okay, this food will be inside a phagocytic vacuole. I told you that if this is the amoeba, if it wants to eat and it sees like a food particle here, do you know what it's going to do? It will now approach it and it will form some sort of pocket. After taking it, after approaching it, it takes it over. Then what is going to happen after that? It's going to seal off. So when it seals off, it enters but as a vacuole. So when the lysosome sees this, it releases the lytic enzymes. They break down that food, which is digestion. They turn it into soluble components. The cell assimilates that. Hope you recall that an amoeba is actually a single-celled eukaryote. So it's representing all other eukaryotes at large. Are we together? Now, autophagy, which I've just explained. When you see phagy, think of phage, think of phagocytosis, think of food. When my tongue starts to oversleep, that means I'm getting tired. But trust me, I'm not giving up. We are completing the table. Hey, autophagy, we have said lysosomes, they secrete enzymes that catalyze the breakdown. You people, you can say at your level, it is okay to say enzymes break down or enzymes catalyze the breakdown. You know that enzymes are catalysts, so you can say they catalyze, or you can just say enzymes break down. Whichever you want, it is accepted. You can say enzymes break down or enzymes catalyze the breakdown. So they are saying autophagy. They are releasing enzymes, and these enzymes can do so many things. One of the things is handling this food. Are we together? And the other thing, they can be working on some other things. For example, a worn out cell, a dead cell, a damaged part of the cell, for example, the mitochondria. Do you understand that if the lysosome is here and the mitochondria is, okay, let's say that is our mitochondrion, and it is here, but it is worn out or dead or it is damaged. So this lysosome can release those lytic enzymes because they are good at breaking down. Then they distort it and break it down. And then a new mitochondrion is formed. And remember, the cell does not have only one. It will always have so many. So if one is faulty or dead, it can be destroyed and the cell keeps respiring. Are we together? That one I talked about it. Now this one is autolysis. Still autolysis, hydrolytic enzymes are produced. Someone may be like, but you told us lytic enzymes. It is okay. Whether you call them hydrolytic, whether you call them lytic enzymes, it is okay. Hey, just because when they say hydrolytic, it's like water is coming in, but it is no problem. Lytic enzymes, hydrolytic enzymes. It is all okay. There's only as to why I explained using lytic and the notes have hydrolytic so that you don't get confused. What are they saying? Lysosomes secrete the enzymes on the whole cell that can cause its breakdown. Are you listening? That the lysosome can secrete enzymes that can destroy it. Destroying the lysosome itself. That means the enzymes can destroy the mitochondria. It can destroy the chloroplast. It can destroy the rough endoplasmic reticulum, the smooth, the nucleus, and everything. So autolysis, we are going to update it and say that on top of lysosomes destroying themselves, even the whole cell can be really affected. Because let me ask, if you have released enzymes that can destroy you, what of other things? So autolysis, we are looking at the whole cell being destroyed. Are we together? Then the autophagy, we are looking at those dead things and even the food that can be really worked on. Okay? And then the digestion, don't really forget it. But we are going to make this better and and tell you again about it the way we used to have it in the old curriculum. You can have the point of digestion alone, so I'll beg your pardon here. Have the point of digestion alone, the way it is here in the notes. Then after, come and look at destroying. Remember the other one was digestion, yeah? So digestion is alone. Are we together? Then also come and look at digestion alone. Sorry, destruction alone. As you're looking at destruction, look at the autophagy. Then be like autophagy, we are just destroying those cells or those organelles that are damaged, worn out, or dead. Are we together? Then look at autolysis as destroying the whole cell. So the lytic enzymes, the hydrolytic enzymes that are being released in this point of destruction, they can, or in this point of autolysis, they can destroy the whole cell. They can destroy all organelles and the whole cell is down. Then autophagy, we are just looking at those cells that are, I don't know whether people have gotten the update. Eh? So I beg your pardon here again concerning lysosomes. The good thing you have you have understood how they really work. So the point of digestion of food, since it is a good point, we shall have it as 1 point. But the mechanism of action is the same. So what I gave the other time was the mechanism of action. So we shall come to destruction. How do they destroy? It is either autophagy, where they are really working on the dead ones, where they are working on the cells that are worn out, or even just organelles that are damaged. Are we together? And the mechanism of action is the same. Hydrolytic or lytic enzymes released, and then those things are destroyed. Then for autolysis, look at it as the whole cell. That yes, even this time round, hydrolytic enzymes or lytic enzymes are what? Released, and they are going to destroy the whole cell. That means the whole organelles are down. So that's what they have been really saying here. So the first point is here, digestion. The second point is autophagy, looking at which kinds of cells, the ones that are worn out, dead, or damaged. What is the other point? It is here, autolysis. Here, what are we looking at? Still the hydrolytic enzymes being released, and it is the whole cell that we are breaking down. Okay? And why is this so important? It is so important that it will help us remove the damaged cells or the old ones, because there is need to replace them by the new ones. Yeah. If at all the cell is damaged or old, it cannot function very well. So autolysis is good that we can destroy the whole cell. Are we together? And then replace it with new ones. And then also this can happen during development, or what we may call embryogenesis. That as we are developing different tissue as we are growing up, there is certain tissue that is not going to be important. So there is need to do what? To discard it. Are we together? Yeah. So that is the whole point. Now there are some examples they are putting here, and we are going to try to distinguish them and see. So autophagy usually disturbs so many students, but I've told you that autolysis, let it be for the whole cell, and let autophagy be for the organelles. Okay? So autophagy, we are releasing lytic enzymes, hydrolytic enzymes, any, which are going to work on organelles. Which kind of organelles? Organelles that are worn out, organelles that are dead, completely dead, organelles that are damaged. Then when I come to autolysis, autolysis is going to work on whole cell, not organelles. Yeah, but it's going to work on the same kinds of cells. I think this is a summary that we really now conclude that. So the whole cell, it can also be a worn out cell. It can also be a damaged cell, or it can be a dead cell. I think people that will be better. So the point of digestion, since it is very good, For it, we give it its own category. Then destruction, autophagy, autolysis. Autophagy for organelles, autolysis for the whole cell, but same nature. Worn out organelles, worn out cells. Dead organelles, dead cells. Damaged organelles, damaged cells. Hope that is now okay. That is the overall summary. Now, this is the very first example. If at all your body is having muscles that are not exercised, remember a muscle is a tissue, so it's made up of muscle cells. If those muscle cells are not exercised, or if the muscle tissue is not exercised, it is not important to the body. So autolysis will happen. All those cells are going to be broken down and destroyed. Because the body is not using them, they are not being exercised, so they are not important. Are we together? So the body will get rid of them. You people have heard of what they call a tadpole. A tadpole, you all know it has a tail. It has a tail, but in the end, after it has fully developed into a frog or a toad, these things don't have that tail that a tadpole had. That means that the tadpole is growing as it is undergoing metamorphosis. That is the period of growth for the tadpole. It will have to lose the tail. And how does it lose the tail? The tail has cells, so those cells have to be autolyzed. How is autolysis going to happen? The lysosomes will have to release the hydrolytic enzymes that are supposed to digest the tadpole tail. And that is an example of what? Autolysis. Did you people know that? That as the tadpole is growing, it has a tail. But eventually when it grows into a frog or toad, those things don't have tails. That means that as it grows, the tail is digested away. Who are responsible for that? Lysosomes. What is the mechanism of action? Releasing hydrolytic enzymes that can digest it away as it grows, as it metamorphoses. Are we together? What else? They are still adding on lysosomes, and they are saying lysosomes release hydrolytic enzymes outside the cell. I told you anything that is exiting the cell, that is exocytosis. It is the mode of transport. Exocytosis. And we can be in position to break down other cells. And the moment you're breaking down other cells, it is like autolysis still, because you're breaking down the whole cell. If it is just an organelle, that is autophagy. Are we together? Yeah. Then we can see this in bone development. But this is now not part of your syllabus. But at least the mechanism of action for the lysosome here, it is very clear that as we are having a bone developing, remember cartilage forms bone. So in that due course, we may need some sort of autolysis to help us with some weird cells that may not enable our bone to really develop so well as we want it. Okay? Now, they are talking of the sperm cell, and I told you already that we are going to meet it. Now remember, this sperm cell is supposed to meet the egg cell or the ovum for fertilization to take place. Is it okay? Now have it in mind that the sperm cell is like this. It's having the oval head. Okay. Then it is having the tail, something like that. So this sperm cell has a structure here called the acrosome. You will learn this. Actually, it's not even on your syllabus because it's under reproduction. But let me give you an idea. It is very cheap. So this sperm cell has this acrosome here. So this acrosome is so important. Remember, for fertilization to take place, the sperm should really get in touch with the ovum cell. Isn't it? You know that. So for them to get in touch, for the sperm to fertilize the ovum, it is supposed to penetrate it. The head of the sperm is supposed to enter the egg cell or the ovum. For those who didn't know the fertilization thing, how it happens for you to form a zygote, the sperm from the male should meet the egg cell or the ovum of the female, but the sperm head should enter into the ovum. So for it to enter the ovum, that means it should destroy this. Remember, the ovum is an egg cell, so it is a cell, so it has a membrane. So the membrane of the egg cell should first be destroyed for the sperm to really penetrate. Are we together? So he is going to help us with the destruction of the membrane where the sperm can pass. It is only and only lysosomes. Where are they? Remember, even the sperm is called a sperm cell, so we expect it to have organelles. So inside this acrosome, the word is here. the sperm cell has what we call lysosomes. So they help us release those hydrolytic enzymes that are going to help us to dissolve the egg cell wall so that the sperm can penetrate for fertilization to happen. Let me hope you've gotten the biology there. Have you gotten it? I don't know if I'm still here with people. Someone is asking which acid, like what do you mean? Okay, so you people are asking. Okay, Achen has something to ask, but please tell me in the chat, have you gotten that thing of fertilization? Do you try to connect it to lysosomes? Sperm cell, on top of its head, it has what we call the acrosome. That acrosome has lysosomes. They release hydrolytic enzymes that can help us dissolve the wall of the egg cell so that the sperm can penetrate and the sperm can fertilize the ovum for us to form the zygote. And that's what they call fertilization. Do you get that biology? Yeah, because you should be in position to also explain it to your friends later. Yes, Achen, you ask, then I go to the next thing.
I'm asking, does the acrosome, does the lysosome that is in the acrosome also facilitate the breakdown of the tail?
Which tail?
On the sperm cell?
Who said the tail goes away?
Yes. Okay.
So the tail does not go away, but the head enters and the tail remains outside. But for you just understand how lysosomes help us with that, because it's the most important thing we need there. Okay? Then this is it. You people know that as a woman is okay, after us forming the zygote, there are a lot of things that are supposed to happen, and then the woman becomes pregnant. But you all know when the woman is pregnant, the baby is called the fetus, and that fetus is attached to the uterus. So for this uterus to really hold this baby for the 9 months, the uterus should grow so much and get so many cells. You get? Remember, the uterus should be highly cellular. It should have so many cells for it to be in position to really hold that fetus for the 9 months. Excuse me. Are we together? Now, when birth is done, the baby has been produced. Now, there is no need for the uterus to be big. It is now supposed to go back to the normal size. That means those many cells that grew on the uterus to enable it to support the fetus should now go away. So we are killing cells. You understand? That is so important. So, are you seeing here? Now here I used the word lytic. So here the lytic enzymes of lysosome, they are digesting away so many cells of the uterus, okay, after birth. And we want it to gradually turn to the normal size. And you people in biology should learn to read textbooks, because when you look at this, we are digesting away so many cells, but all those cells are belonging to the uterus. There is a book that calls that autolysis. There are 2 books calling it autolysis, and there are 4 books calling it autophagy. That's why they say that if we are marking students at A level, let's go with autophagy, because it is 4 books saying that. But at least what you should appreciate is the mechanism of action. You're not supposed to say autophagy. For you're just supposed to tell us how the lysosomes are going to help us digest away those many cells of the uterus that are not needed after birth, so that it goes back to its normal size. Is it okay? So that is it. That is it. Where confusion comes in, there are some terminologies you can leave out. Now, in case what we are digesting is bacteria, because bacteria can cause infection, we are doing body defense. Are we together? In case what we are killing or destroying is bacteria, and bacteria can cause infection, that means what we are really doing is defense. What is body defense or defense of the body? Something like that. Okay, now that is the lysosome for it. It is very serious. And that's what really makes eukaryotic cells really hard. Talking about each of these organelles one by one, one by one, one by one, one by one. and it is very serious. I want to tell you that point blank. But we always want to have a session where we are going to explain everything. Whether people are here, whether they are not here, we are going to complete. Okay, now let's go to the nucleus. The good thing, you know already how the nucleus looks like. So as I talk about the functions, I'm going to be connecting it to those different components of the nucleus. But before we really go through it, I think someone has something to say. That is Devi. Please, what are you asking, Devi? And be fast.
I'm asking doctor that are the lytic enzymes acidic or alkali?
You don't
Yes,
need that
doctor.
content
Okay.
because the pH of the media for the enzymes depends on what you're destroying. So for you, what you
Okay.
need is simply lytic and nothing else. Okay. Nucleus. It contains genetic material in form of chromosomes, you know that? And you know that genetic material, if at all it is transmitted from the parents to data cells, that is transfer of traits, and we can have inheritance, isn't it? And you all know the nucleus is the control center for all cellular activities. Who doesn't know that? Okay. DNA is organized into genes, and it is those genes that control activities. But if that is very hard, just say it is the control center, or it controls cellular activities, period. But if you want to learn more, say DNA is organized into genes. And I explained that in nucleic acids. So I don't want to overstrain myself here. Just have it as simple as the control center for cellular activities. Is it okay? They carry instructions for the synthesis of proteins. Now, protein synthesis is a topic, and we shall see how the nucleus participates in the protein synthesis. This is a full topic, central dogma. I cannot even risk and start explaining. But we shall see that in protein synthesis, how the nucleus participates, and you will understand. Okay? Involved in the production of ribosomes and RNA. And this is because of the nucleolus. Yeah, I told you that I will tell you about the nucleolus. Those who are good at reading textbooks, you will find that inside the nucleolus, there is what they call the nucleolar region. So that nucleolar region of the nucleolus is the actual site where these ribosomes are actually what? Produced. Then they later go and be suspended in the cytoplasm. But all those things will come, especially under nucleic acids. I told you in the lesson of cytology, the first one, that Na means nucleic acid. So anything that involves Na, nucleic acid, when you don't get it so well, you wait for nucleic acids. Everything is just explained well. It is a topic of term 2 here. Let me show you something here. Term 2 work, inheritance and evolution, nucleic acids. Do you see them? There is lesson 1, lesson 2, lesson 3, and lesson 4. So when you watch them, all these things I'm saying they will just become so cheap for you. So time will come when those things really make sense. Yeah. Involved in cell division. Who doesn't know that? Yeah, even in cell division. So when you say cell division and you cannot really connect about how the nucleus is participating, I now refer you that you're supposed to watch that. But cell division, have it in mind, it is the nucleus that divides first, and then the cytoplasm cleaves, what we call cytokinesis. So it is the nucleus that really helps us with those things. Remember, for us to divide, DNA must replicate, and DNA is inside the nucleus. So it should participate as a cell is what? Is dividing. Now they are talking of a Golgi body. Now this Golgi body, I told you so many things. Have it in mind that this Golgi body is just good at secreting. It is just good at secreting. If you can understand this, well and good. Well and good. So for it is just good at producing things. Even here they are telling you it is producing glycoproteins. In the future, you will see how glycoproteins are produced. That is in the next lecture to do with the cell surface membrane. And then I'll also remind you that I told you that glycoproteins are produced by the Golgi apparatus, and this is how it also does it. Are we together? Now they are telling you that the very most common glycoprotein that you should know as a senior 5, senior 6 student is mucin. Mucin is a glycoprotein. When you see mucin, what really comes to your mind first is mucus. And mucus is so important in biology because we have it in the respiratory tract, we have it in the digestive tract, we have it everywhere. The digestive tract is what we call the gastrointestinal tract, from the mouth straight away to the anus. We have mucus lining the walls of all those structures, the intestines, the esophagus, the stomachs, and so many things, okay? So it is required in so many secretions to an extent that it is mucin that really helps us to have the mucus to be formed. Are we together? And all of you are here, you know that mucus is so important for lubrication. If you don't have mucus in the esophagus, food cannot be easily swallowed. Otherwise, food would be stuck in the esophagus. But because there is mucus lining the esophageal wall, food can really be lubricated and it just goes, the bolus. It is so important for protection. How? You know that the stomach wall has mucus. So because of that, that's why even when you have acid in the stomach, you may not be affected so much. Because that mucus that is lining the stomach wall can really protect it from acidic effect. Then pathogen trap. The mucus you're having in the respiratory tract, the mucus you're having in the digestive tract, the mucus you're having anywhere, even in your nasal cavity, it can trap bacteria. It can even be by God's grace and it captures Mycobacterium tuberculosis, and then you don't get TB. But if it's not captured, you find yourself getting tuberculosis, something that is really very bad. Are we together? So pathogen trap, protection, and lubrication. Now I've given you an example for the esophagus for lubrication, protection for the stomach wall, pathogen trap for the respiratory tract. And they are not here, but I've said them. So you're supposed to write them so that as you're reading, you don't ask yourself that how are these things really connected to mucus? Then concerning secretory things, I told you these things. Golgi body and secretions are just like this. When you secrete pancreatic juice, you're having what? Pancreatic enzymes within it. When you secrete the saliva, you're having salivary amylase there. So each and every digestive juice has enzymes there. So if you secreted juice, you have actually secreted what? Enzymes. It is involved in the formation and repair of membranes. Do you remember when I told you that membranes have glycoproteins that may work as signalers? I told you about that. And I've just told you that Golgi apparatus can even produce glycoproteins. So do you see that really they can be involved in formation and repair? And we shall also see that when you reach cell membranes, what are those things that make it up that the Golgi body can provide? And if it can help us with cell walls, why would it really fail for cell walls and insect cuticles? Oh, those are protectings. You understand? They are protective covering. So they can really, the Golgi body can really help us. The rule is simple. These cell walls, cell membranes, insect cuticles, they need certain compounds that really make them up. You hear things of cellulose, peptidoglycan, chitin, a lot of things. So the Golgi body being secretory, it can secrete those things and they are used to make those things. Are we together? Then another thing is that the Golgi body can form lysosomes. Someone can be like, why? Remember, we have said it can secrete enzymes. So if at all the enzymes being secreted are hydrolytic, and then those hydrolytic enzymes are now covered by a membrane, this one becomes a lysosome. Because inside there are hydrolytic enzymes and then a membrane around. I remember telling you that the Golgi body gives off Golgi vesicles. Now, if the Golgi vesicle going off has inside hydrolytic enzymes that have been made by the Golgi body, that means it has become a lysosome. Then also these lipids and steroids. I remember talking about steroids in the, as I was giving you research work on lipids. Steroids are just a derivative of lipids. In simple terms, I mean that steroids come from lipids. So when I say steroids, I mean lipids. And we shall see what these steroids really are when we have that premium class on lipids. So it's not really bad. Someone may be like, how is really the transport happening? Are you seeing this Golgi body? It's like this, eh? So transport really happens until the Golgi vesicle is pinched off. Remember I told you there are interconnections. So there is that transport that happens until the thing goes out. So the transport can be intracellular inside the cell, and it can also be extracellular. Because this thing is giving off as a secretory vesicle, it can then later move out of the cell by exocytosis. I told you exocytosis is exiting the cell. So whatever you're giving off can later on exit the cell. When it exits the cell, that transport is now extracellular. When the transport is just within the cell, it is now intracellular. And there are organelles that cannot do the extracellular, like the endoplasmic reticulum. Remember, for them, they are just connected to the nuclear membrane. So for them, they can only do transport within the cell. They cannot reach that extent of pushing something to the outside. And we're also going to see it here. Then I told you that strictly plant cells, they are Golgi bodies. They are so special and they are called dictyosomes. And Golgi bodies or the Golgi apparatus or the complexes, they are common in secretory cells. So that makes the chief function of Golgi bodies being secretion. So you take note of that. Are we together? So that is the Golgi body for you. So we have the rough endoplasmic reticulum. Reticula is plural. Then reticulum is the singular. I hope you know that. So rough ER, they can abbreviate it like that. Then you also have the smooth ER. They can abbreviate it like that. So you all know that what makes this rough are the pimples. They are the ribosomes. And no ribosomes are just good for making what? Proteins. But don't forget that they have those cavities they called cisternae. So transport can occur within them, just like the cisternae for the Golgi bodies. Are we together? So have it in mind, they are cavities, cisternae. So transport can really occur there. So they are telling you that their cisternae can have transport. And I told you that transport is strictly intracellular within the cell. Why are they transporting? They could be digestive enzymes, they could be hormones. You understand? And these ones are made by strictly the ribosomes. Remember, the RAF has enzymes on it. Sorry, the RAF has ribosomes on it. And ribosomes make proteins. And proteins can be hormones or enzymes because both are protein in nature, but not all. Are we together? So, nah. So that's what I'm saying. That the enzymes and hormones are now examples of proteins. And in proteins under chemicals of life, we shall see that they are called functional proteins. Okay? And there are some of those ribosomes can make. Are we together? Yeah. And when ribosomes make them, since they are on the rough endoplasmic reticulum, it is the cisternae for the rough endoplasmic reticulum to do the transportation. And it is strictly intracellular within the cell. But the Golgi body is so special that for it can go as far as extracellular is concerned because of being in position to do or to form secretory granules that can later move outside the cell by exocytosis, as we saw in the diagram of the animal cell, the BS black and white, if you recall. They are saying that the rough ER also forms part of the cytoskeleton. I told you something, that the cytoskeleton, so I put here for you the food for thought. And I already said it, so whatever I'm going to read here, you know. Apart from a few things, those are the proteins. And I'm saying the cytoskeleton is a network of protein filaments. What do? They give the cell shape, they give the cell support, and also aid in movement. Are we together? And I told you the cytoskeleton is made of three things. And they were apart, and you saw them already. All of you saw the microtubules, all of you saw the microfilaments, all of you saw the intermediate filaments. So the microtubules, because of the word tubules, this one, tubules, the protein that is made up of them is called tubulin. Then the microfilaments, the protein that makes them up is actin. That's why I told you that I will tell you later on why microfilaments are also called actin filaments. It's because of the protein actin that makes them up. Then these intermediate ones, they are intermediate. For them, the proteins are so many. And I just gave you one example, that is keratin. So these are proteins making up the components of the cytoskeleton. But who is going to synthesize these proteins? It is ribosomes. And where are these ribosomes? On the rough endoplasmic reticulum. So do you see how it is indirectly part of the cytoskeleton? That it can give you proteins like tubulin, actin, and keratin, which make up components that make up the cytoskeleton. Hope you see the connection. Important in synthesis of the cell membrane. Do you remember when I told you the cell membrane has glycoproteins? It even has proteins without even being glyco. We shall see them. And ribosomes can do protein synthesis, so it is important. Okay? It provides a large surface area for metabolism. Protein synthesis falls under what we call anabolism. I talked about it in the very first class of cytology. And I told you anabolism is biosynthesis. And protein synthesis is a form of biosynthesis. Biosynthesis, you're simply synthesizing biological compounds or components or substances. And the proteins, of course, are biological compounds. Are we together? So if metabolism, which is anabolism, which is protein synthesis, can occur on ribosomes, on rough endoplasmic reticulum, then it is a large surface area for metabolism, or it provides surface that is large enough for metabolism to happen. Hope you get the connections. So I don't want you to cram. You can cram, but please to understand the connection. So that even if they set a scenario, the words I say can really run through your mind and you'll be like, oh, this was said by doctor, so this is it, and that is it. Okay? So smooth, we have talked about the rough. The rough, you have realized that what dominates everything, it is the ribosomes. It is the ribosomes. So you've been talking about proteins, but these ones, they are smooth, they are no pimples, they are no ribosomes. You understand? But these ones are known for lipids, transport and synthesis. I told you if lipids are synthesized, even steroids will be synthesized. You understand? And the chief function is that. But remember, they also have those cavities, so even transport is very okay. Are we together? So lipid synthesis, lipids can never leave steroids, they are relatives. Are we together? Then they play a role in carbohydrate metabolism. Someone may be like, how is that even really coming about? You all know that there is when lipids can be converted into carbohydrate, and there is when carbohydrate can be converted into lipids. You understand? So all that is carbohydrate metabolism. Whether you're turning carbohydrates into lipids, whether you're making carbohydrates from lipids, that's all metabolism involving carbohydrate. And since the smooth is just the friend of lipids, anything involving lipids can also involve other things. You understand? And converting lipids or synthesizing lipids is a chemical reaction. So when we say that it can also provide a large surface area for those reactions like biosynthesis or synthesis of lipids and steroids to happen, then it can be a good function. Okay? Then the other thing it helps is it also contains some of the lytic enzymes. Yes, you will forgive the biology subject because some of the things are facts. So they don't even have an explanation. They just tell you the smooth has lytic enzymes. They will not tell you what it has. It is having, because they made research and microscopy and they found out that the thing is there. The enzymes are there. And they are even attaching that the lytic enzymes are strictly in the smooth ER of the liver cells. And the liver cells are what they call the hepatocytes. You'll find that in homeostasis of FA. They liked that word so much, hepatocytes. So their smooth ER contains lytic enzymes, and you all know what lytic enzymes can do just from the knowledge of ribosomes. So because they also have those cavities, they can help us with intracellular transport. I told you they not only synthesize lipids and steroids, but they also transport them. So for you, if you think smooth ER is hard for you, help me and capture two things. Have it in mind, it is a friend to lipids, and lipids never leave steroids. They not only manufacture them, but also transport them. So you have 4 points. You have manufacture of lipids, intracellular transport of lipids, manufacture of steroids, intracellular transport of steroids. Hey, That's how they used to cheat students of old curriculum, and they used to get 4 marks. Because you're talking of lipids and steroids, different things even when one is the derivative of the other. Okay? And since we are making lipids, and I told you that the membrane has those phospholipid bilayers, it has even glycolipids. So lipids are needed to be part of membranes. So when they say that they provide structural support, it is very correct. Because what provides the support are membranes. So if membranes need lipids and Smoothie Ara gives us lipids, that means they are keen providing structural what? Support. And remember, it is even around the nucleus, so it can provide the support to the what? Nuclear structure without even going through the production of lipids that are needed to be part of the membranes that do direct protection. Are we together? Hey, and thank you for staying on, those who are on. But let me try to check people because it's now 1 a.m. I don't know if people really are sober. I want you to write for me this number in the chat if at all you're sober. Yeah, you write for me this number in the chat so that I see the number that is on and the number that is sleeping. I'm not always supposed to end class when people go. Even when they run out of battery data, I'm supposed to keep teaching because we keep this work. Even you are here, you're tired. That means you need to review this work. So when I get a chance and I can speak, I will speak until I complete. After all, we are doing this for the future. Even in the future, you can get back and do what? And look at this. You see, the people are many, but those who have written the number are very few. But I'm happy that people are at least 10. Yeah, at least people are 10. That is not bad. So the 10 people, yes, we can push and represent the rest. If anything is not clear, you tap me. If I start talking uncoordinated statements, you tell me, doctor, you're tired, you sleep. So let me hope I'm still on form. Hey. We go. Sap vacuole. I told you about sap, that sap is any fluid, any substances, provided it is being transported in plant tissues. That's why you can never find the sap vacuole in what we call the what? The animal cells. So, since it is just going to store, remember the components of sap are supposed to be transported. So if it is to store, it is supposed to store them temporarily because they are supposed to be transported. That is the statement we said. So whatever it's going to store, it is supposed to be temporarily. The food nutrients, the sugars and amino acids temporarily. It can also store the pigments that can that produce different colors in different parts of the plant, like flowers and fruits, and this can attract pollinators and dispersal agents. This is how they are connecting content. That yes, the food nutrients are being, there are so many, sucrose, glucose, amino acids. This one is very clear. Here they are saying that even the pigments that give flowers color or that give the fruits color, those pigments can also be stored inside the sap. So they are saying when it stores them very well and later releases them for them to be used, the flowers that are going to be given a very good color will attract pollinators. The fruits that will have a very good color, they will attract dispersal agents. Do you know that when any animal eats a fruit, like maybe an orange, and then it defecates somewhere, the seeds of the fruit are indigestible, like they are not digested. So wherever it defecates, you can have an orange tree growing there. Do people know that? Look at the seed of the mango. Look at the seed of the avocado. It's indigestible. So the moment anything like the hippopotamus or anything gets that fruit and it eats it, the seeds like those of the watermelon, they are very hard. Those of the pumpkin, very hard. They cannot be digested. Once defecation happens, trust me, they will grow back. That's a form of dispersal. Then attracting pollinators, that is very cheap. What else do they temporarily store? They temporarily store organic wastes. And of course, you know that. Organic wastes, the moment they accumulate, the plant just gets rid of the organs. Did you know that? Plants don't joke. The moment any plant organ gets too much wastes in the vacuole, the plant just gets rid of the organ. That's why you see leaves falling off. Yeah, That's why you see a plant having very weird roots. It becomes very weak and it even falls. For it just gets rid of the total organ. It doesn't mind about the cell. Okay. It can also temporarily store the hydrolytic or the lytic enzymes, and they also perform similar roles as those of lysosomes. So for the moment you see lytic enzymes or hydrolytic enzymes, think of the other things. Digestion, which is very nice. Destruction, look at it as autophagy for organelles, and look at autolysis as the whole cell, both of which organelles and cells are looked at as worn out, dead, and damaged. Are we together? There is a topic they removed from your syllabus, but it was so important. But I believe you've talked about, or you talk about these things in the practical of biology. Remember, this sap is having a lot of solutes. Yeah, solutes, we mean those salts, maybe amino acids, they can be sugars, they can be anything. So they make the cell to be too concentrated. So another word for concentrated is hypertonic. If you don't understand the terms of hypertonic, isotonic, and hypotonic, I will discuss them sometime in biology practical. Don't worry. But have it in mind that concentrated is synonymous to hypertonic. So if the cell is too concentrated, it will need water. Because when it becomes too concentrated, it may die. So water should now be absorbed. And of course, water is absorbed by what? By osmosis. After all, we are having even a difference in concentration what? Levels, or even have a concentration gradient. Are we together? So when that water is absorbed by the concentrated cell that is about to die, so it's fighting for its life, you all know that that water that is absorbed will bring about hydrostatic support. How? The cell will become turgid, and when it becomes turgid, it becomes rigid, and rigidity means providing support. Which kind of support? Hydrostatic support. Though here they have specified that herbaceous plants, but even other plants, but mostly the herbaceous, or the ones they call the non-wood. Are you aware that those herbs don't have wood? Yeah, if you've looked at herbs like dodo, it is non-wood, it is herbaceous. So that support it really gets, it's because of the turgidity that comes from osmotic absorption of water. You understand? So that sap can really push it, making the cell concentrated, making it to urge more, making it more thirsty for the surrounding water. It gets absorbed osmotically, and things happen. Yeah, we are in the evenings of the class. We are about to finish. Chloroplast for photosynthesis, everyone knows that. Ribosomes, protein synthesis. Who doesn't know that? We all know they may be attached to the ER, or they are free in the cytoplasm. We also know the chloroplast and mitochondrion, they also have ribosomes. Those are things we explained. When you see RNA, I told you that that one will be understood when we reach nucleic acids. Yeah, but of course, you should be knowing that this is ribo. Even this R means ribo. So that means they are connected. Yeah. So that means there is future of understanding. Then we are saying, and I also said in that protein synthesis and nucleic acids, that these ribosomes can be a group. They can collect themselves and be together as a cluster. So when we have a group of so many ribosomes that are strung together, they call it a polysome. If you don't call it a polysome, you call it a polyribosome. Remember the word, when you see the word poly, they are meaning many. I think you people know that. From mono to di or bi to tri to tetra to pent up to when you reach poly. So many ribosomes, polyribosome, or just polysome. And who is joining them together? It is the mRNA. And we shall see that in nucleic acids for you to understand that part of by mRNA. But of now, at least appreciate that a polyribosome, there are many ribosomes, and you can either say polyribosome or just polysome. Are we together? Hey, we go. So we also have storage granules. So storage granules in the eukaryotic cells are so many. There are those that are storing carbohydrates as glycogen, so we have glycogen granules. There are those that store carbohydrate as starch, so we have starch granules. So they are saying for the plant cells, we are having starch, and starch is stored in specialized structures called amyloplasts. Remember when I told you about amyloplasts, which are an example of leucoplasts, and leucoplasts are an example of plastids, and plastids are only present in plant cells? Do you recall all that content? Remember when I told you that I didn't want to say it at that point, I wanted it to come this side? So, all that is work. Content. So the glycogen granules, of course, they are storing carbohydrates or sugars in form of glycogen, and it is the fungal and animal cells. Then for the lipid globules, I told you they are either globules or droplets, and they are also found there in the cytoplasm suspended. The way you hear it, lipid droplets or globules, they are just there to store lipids. Nothing much. Okay? Then we go to the cytoskeleton, which you know very well. It has the microtubules, it has the microfilaments. Okay? I told you that microtubules, what do they have? They have the protein tubulin because of tubules. So the protein that is present here is tubulin. Are we together? Then the one that is present here in the actin, in the actin filaments or microfilaments, it is actin. And I told you when you see actin, think about what? Contraction. So let's see. So it is a skeleton, don't forget. So it being a skeleton, it can give the cell shape. It being a skeleton, it is key for movement. You see such things? I told you that endocytosis and exocytosis, it is movement. When you see exocytosis, you are exiting the cell. Endocytosis, you are entering the cell. And that is movement. And movement and skeleton, they can be in the same sentence. You understand? If a cell is like this, then it becomes like this. Without the cytoskeleton, okay, it is the cytoskeleton to help us with all this. And for those things to happen, the cell should be, the cell surface membrane should be modified somehow. And without the skeleton to give you that flexibility, the movement cannot happen. Hope you understand. So connect the cytoskeleton to how flexible the cell becomes when movement across it is happening. Okay? They are telling it is associated with cellular movement because it is because it is a skeleton. So with cellular movements, that's not a surprise. Intracellular transport within cells. Even the cytoskeleton is within cells, so it is very important. You understand? Remember these things that are made up of it, They are saying microtubules. You people, microtubules, they are tubes. If you go back to the structures I've been showing you, the microtubules were like this, Tubules. Tubules means a small tube. Just venules mean a small vein, and arterioles mean small arteries. You study that in transport in human. So when they say tubules, they are small tubes. So they are still tubes. So them being tubes, do you think they can really fail to provide a route? Can they really fail to provide a route? They can't. So providing a route for the transportation of materials is very okay. So you may ask, how do really Golgi vesicles reach the deposition sites at the cell membrane? It is because of these tubules. But remember, they are micro. You cannot see them with your naked eyes. You're supposed to first have the microscope and then see how they are helping us to move those vesicles from where the Golgi body has given them to the cell membrane. Are we together? Hey. They are a major component of the cilia and flagella. If you know that cilia and flagella the cilia and flagella are responsible for movement and locomotion, then you cannot be shocked if at all they are connected to the components of the cytoskeleton, because it is all to do with what? With movement. They have said, I seeing all this content. This content is to do with cell division, because I know you people have heard of what they call spindle fibers. So we shall see in cell division how microtubules come in. We shall see that. So don't worry. We shall see in cell division how the microtubules do what? Come in. So don't worry. And how the cytoskeleton. Are we together? Yeah. But if you know that, if at all you know that stage they call anaphase, that involves movement of chromosome to opposite poles. So they are moving. The moment movement is happening inside the cell, you cannot convince me that the skeleton is not contributing something. There should be something the skeleton is contributing for that movement to be happening. Are we together? So you cannot be shocked. Those have been the microtubules for you. What of these filaments? I've told you they are called actin filaments. So when you see them, you think of what? Contraction. It is even here, contraction, so you cannot be surprised. But since they are components of the cell skeleton or the cytoskeleton, support and maintaining shape cannot be a problem. Don't you know that? That the skeleton, being a framework, is there ready to support us and maintain our shape? You know that. Okay? Now, I've told you that contraction and constriction don't really differ so much. Contraction and constriction, even when you just ask Google right now, they somehow mean almost the same thing. Okay? So we shall also see this, but they are talking of cytokinesis, which happens during cell division. So as I told you that this topic, it was a disclaimer. It has a lot of ideas from so many topics. Some of the points I'll be advising you will wait. Then those where I can say something small you can understand, I've already said it. Okay? Then cell motility, don't forget. A cell can move by what they call cytoplasmic streaming. Okay, cell motility. And I told you for the cell to really be flexible to take up the solid food, to take up the drinks, that flexibility that enables it to form that cup pocket to take up the food, the drink. The cytoskeleton should give us that flexibility. Because if it was very rigid, maybe the cell could not even fold to make those invaginations, to make those indentations for the food to be taken up, for the drinks to be taken up. So cell motility, those things of phagocytosis, pinocytosis, they are just so important. And this is cell eating, this is cell drinking. So the cell is taking in things. And in the future, I'll tell you that these two things are forms of endocytosis. Endocytosis means that things are entering the cell. What is entering can be food, phagocytosis, or a drink, pinocytosis. Forming phagocytic vacuoles for food, forming pinocytic vesicles for the liquid. Okay? Hey, you cannot tell me that you're more tired than me who is speaking. For you're just listening. Hey, and I don't see whether you have any reason to doze. That one is completely optional. Okay? So we're having centrioles. I told you about these centrioles. These ones on right angles. Right angles. Those ones, right angles like that. Okay? So they are telling you that these ones, within them, they have microtubules. So the mere fact that they have microtubules, you cannot be surprised if they have some of the components of microtubules. That's why for me, I just usually advise students to just have that point only. Say they form microtubules that make up the cytoskeleton period. That means you will have pulled off the functions of the microtubules which I've given you, and also those of the cytoskeleton generally, which you also already have. So it's like they are hiding in microtubules and what? And the cytoskeleton. Are we together? That is something that is very cheap. Now, when you see these peroxisomes, I told you about them already, and I told you they are the ones we call microbodies. So when you see peroxy, as a student of biology, it is 99% chances that you do chemistry. So the chemistry thing that comes into your mind is peroxide. And when you write peroxide, the very first peroxide that should come at the back of your mind, the first one should be hydrogen peroxide, not sodium peroxide. So when you see peroxisome, think of hydrogen peroxide. And the very first thing I want you to be recalling about hydrogen peroxide is that it is very toxic. Very toxic. It is 1:22. I'm supposed to be at the hospital at 2 a.m. That means I pray that we finish on time. Yes. So let's see. I've said that hydrogen peroxide is very toxic. So when you see peroxisomes, understand they are micro bodies. Then be like peroxy, I think of peroxide, and the peroxide is hydrogen peroxide, and it is very toxic, even the way it sounds. So because it is very toxic, maybe these peroxisomes are going to help us break it down. Because hydrogen peroxide is very dangerous to the cell. So the toxic hydrogen peroxide, these peroxisomes can help us to break it down. But remember, the only enzyme that can help us to break down the hydrogen peroxide is called catalase. So catalase enzyme can convert the hydrogen peroxide into water and oxygen, like that, water and oxygen. Hope you're okay with the chemical formula of water. I taught that. Even oxygen, everyone knows it is O2. So how does hydrogen peroxide look like? It looks like this, H2O2. So it is very toxic. Catalase can help us to convert it into water and oxygen. These ones are not toxic. So do you see how it serves us? So someone will be like, where is this catalase? This catalase is inside the microbodies. So for it just waits for this hydrogen peroxide. The moment it sees it, it breaks it down because it is very toxic. So it will form water and oxygen that are not so toxic. Are we together? So where is this hydrogen peroxide coming from? It is a metabolic waste. By metabolic waste, I mean every time metabolism happens, chances are high that some of the wastes that are going to be as a result of this metabolism, it is the hydrogen peroxide. So don't think they just maybe give it to you as caliform. No, it is when metabolism happens, things like that. So the catalase does not stop on that. It can also help us to break down fat to form what? Sugars. But the most important thing is that the other things come in later, like this glyoxylate cycle. It is not so important at your level. And one of the things I'm actually giving as research, you can make research about it, but they will not set it. It's not your level. Even when they ask you roles of or functions of the microbodies, you cannot spell this word correctly in the ANEC exam, but you end up writing something else. But you just go to YouTube, put in glyoxylate cycle. Then you choose a video of like 5 minutes. You look at it and be like, oh, this is where the catalase is coming in. The hydrogen peroxide is here. Things like that. But the best thing is toxic hydrogen peroxide into the less toxic. So it's better you say less toxic, or you can say non-toxic, the water and oxygen, because that oxygen can be toxic, yeah. But you study that at higher levels of education. So I think less toxic is better. Hope peroxisomes are very okay. Now the flagellum, this one we talked about it in prokaryotes. It is responsible for, let me get the notes again, but it is just responsible for locomotion. Even the cilia locomotion, who doesn't know that? You know the sperm tail. It is a modified flagella. So it is also important for the sperm to swim. If you really want to see the sperm swimming, it is so important. Are we together? So never take that for granted. So sperm cell, it has a tail, but that tail is a modified flagellum. So it can also be in position to swim towards the ovum so that fertilization can happen. Even in the Euglena, which is eukaryotic protoctist. Are we together? And also the trypanosome, protozoan. If at all you were good at primary science. Cilia, what is all with this cilia? So we know that cilia can beat rhythmically. You recall that cilia? I remember we watched the video, you saw the cilia, you saw the flagella. Though the other time it was pili, but the pili is like the cilia. The pili you saw in the prokaryotes video, it's like the cilia. So it can also beat rhythmically. But you can find that this cilia is on the oviduct. Do you people know how the female reproductive organ looks like? Let me just draw a sketch so that I tell you something. So this is where we have the ovaries, yeah? So ovaries are the ones that are going to give us the ovum. Do you understand? So when the sperm comes, it should go up to where the ovum is, and then we shall form the zygote from somewhere here. So all this area is the oviduct, somewhere here, where the fertilization takes place. So the oviduct has cilia. So that cilia is supposed to beat rhythmically so that it can propel the zygote, so that it can push this zygote to come and be here in the uterus so that it is implanted. And when it is implanted, do you people know those things I'm saying? Remember, the zygote, it's supposed to be implanted here on the uterus. That's why it develops into the embryo, fetus, things like that. So, fertilization takes place in the oviduct. The sperm must go up to where the ovaries are in the oviduct, somewhere there, to meet the ovum to form the zygote. Now, this zygote is supposed to be pushed to go to the uterus to be implanted, so that it can now grow into the baby. Are we together? So if at all the oviduct didn't have cilia, maybe the zygote would be hardly propelled. Maybe it would even be implanted in the oviduct, something that is very bad. I don't know if you've heard of ectopic pregnancy. When you have implantation anywhere else that is not the uterus. Are we together? Then the cilia can also be rhythmically in the respiratory tract. Even your nose has cilia. Therefore you call it hair. But the moment it keeps beating rhythmically, it can really trap the foreign materials and even expel them, like the mycobacteria, tuberculosis, that may bring TB. Okay? In paramecia, instead of the pili in bacteria, for it has the exact cilia. Yes, this was a full topic or a subtopic in the old curriculum. We could also be covering those things, how it really locomotes. It's really very nice, but it is absurd, it can't happen. Then also at the paramecium, this paramecium, it has a mouth like this, or what they call the oral groove. Now this mouth has some cilia here. So in case this is food, and the paramecium has recognized that food, this cilia should also do the same. It should try to draw water currents and push this food into the mouth. So the cilia for the paramecium, it not only helps locomotion, but also it is good for drawing water currents containing the food it wants towards the oral groove or the mouth of the paramecium, and that is very key for feeding. And you should also be recording these sketches so that they can also help you in bringing back the content to life when you need it for an exam. So I congratulate you all. We have finally reached to the end of the table. Thank you on for everyone who has stayed on, even when you really felt tired. May God bless you so much. Yeah, but God also needs to also bless me because I've also stayed here. Okay, so they are also good for detecting stimuli because you call them hair, the cilia. So that means they also have the sensory hair cells. So because of having that property of being in position to sense, they can help us detect stimulus. If the stimulus is food, they attack it. If the stimulus is toxins, they run away from those toxins. So detecting this stimulus is very key for survival. But we shall add on these when we are revising scenarios, and I teach how best these things can be really explained. But you've really gone with a lot of content. Yeah, but at least I'm happy that things have been explained. Now, I'm the same teacher who taught you prokaryotes in the previous class, and in this class I've taught you eukaryotes. So the comparison, it is supposed to be your research, but I'm going to allow you to take screenshots for that research. Yeah, I'm going to allow you to take screenshots for that research so that you build on it. You just make sure whatever you're reading is correct. Whatever you're reading is correct. Whatever you're reading is correct. What does not make sense to you, I've taught you so many times on how you can really make your research. Because trust me, as I compare the two, everything I'm going to be saying is going to be a repetition. Something which is very bad. It's going to be a repetition. So I always want people to make notes from these videos. So if you've not been taking screenshots very well, I really pity you. So you all know that this cell surface membrane, I'm not really going to give this to you as your research. It is going to be our next stop. I'll teach it, but it will be a premium class. It may not be on YouTube, maybe in 2030. Because now I've taught you a lot and I would want you to go back and revise this work and really understand it. So this will be a premium lesson. Okay? Then the other thing that I want to really tell you about, I'm really feeling bad that I didn't use this diagram of the Golgi apparatus. We told you that the Golgi apparatus is good at forming glycoproteins. I'll tell you that glycoproteins, glyco is for carbohydrate, then protein. Are we together? That when they say glycoprotein, for glyco, they are meaning carbohydrates, then also the protein part. Are we together? Now, for the Golgi apparatus to make a glycoprotein, it needs one of them. It just needs a protein. Then it's the one that really adds the short carbohydrate chain, but we shall see that in the cell surface membrane. But I just wanted you to understand that when they are making a glycoprotein, it's not that they are making the whole of it like it. No, glyco is for carbohydrate. That's why they tell you glycogen is a carbohydrate. So it needs either a carbohydrate first, then it later modifies that carbohydrate by adding a protein to make a glycoprotein. But in most of the cases, it is the protein first. So then they add a short chain carbohydrate. So that short chain carbohydrate, when it conjugates or when it gets attached or added to the protein, that's how the glycoprotein is formed. Same to the glycolipid, that it is a lipid, but they add a short branch or a short chain of carbohydrate or a short branch of carbohydrate, and then it becomes a glycolipid, because even glycolipid is carbohydrate and lipid. So it is a lipid, then they add what? So the glyco or the Golgi, I mean the Golgi. You people, Golgi was a person, Yeah? Golgi was a person. So this is supposed to be strictly and strictly capital G. I don't want to see people who are writing small g for Golgi. It was someone, so you respect them. Is it okay? Now, yeah, that's what I wanted. So that's how the Golgi body does those things of glycoproteins and lipids. It has a protein first, then it adds a short chain carbohydrate to make a glycoprotein. It's having a lipid first, then it starts to transport. Imagine it starts from here. So this is how things move. Do you see? And finally, just because I don't know why your curriculum does not want us to explain the structure of the Golgi body alone. And then I talk about the outer surface, the inner face. I don't know why, but those things were really nice. But at least we have really tried our level best. You know the sketchy structures, you know the colored ones, you know the ones of BS black and white, you know the functions. And that is the best thing, because they want you to be applying this content. So you knowing the function is very key. And of course, some little bit of structure. So we shall really make this content of cytology stronger if at all we do what? We shall make the work of cytology stronger when we start revising it. But please, the assignment I'm going to give you is revising this work. Is the most important thing. I want to be okay. So let us take screenshots. Very fast. Okay, you can take that, but you be fast. Those who have been taking, even if you don't take. You take that screenshot and tell me if it is clear. You first take it and check it. Tell me if it is clear in the chat. So I want people to leave this class when they are not really the same way as they came. Is it clear or not clear? Because again, taking screenshots that are not clear, okay? People have said clear, okay. That's why I always tell you that in the end, you're supposed to share these screenshots to your friends in the group, say, because someone is saying not, another one is saying clear. So you take that also. But you hurry. I need to be at the hospital at exactly 2. Maybe I'll push my shift to 2:30. Provided I leave the class when I know it has been a success. then you will be a happy man. You take that also. You take that also. If I have a tendency of saying I don't give them my beautiful notes, what am I doing right now? I'm giving you very good work. You take that also. Be fast. Okay, then we go here. Then you say I have bad manners with these very beautiful notes. You take that. Just because we lost the other one that had the writings in it. But if you've been following, you can really also make yours dirty like that. But please read today's work. To me, it has been too much. Hope you don't change combinations. Biology is sweet. Go here. Okay. The data work is here finally. But it came at the end. You see now, this is the comparison. Yeah? It is research, but I'm also going to give you the notes. You go and read and make sure whatever you're reading makes sense. Where you don't understand now, you ask. That means if you don't understand something on the side of prokaryotes, that means you didn't understand the prokaryotic work. There are so many differences. The other one was almost a full page. Even this one is a full page. Yes, there may be repetitions, but you will find out. I cannot really teach you all that. I cannot really teach you all that, and I also teach you the comparison. So, you also take that last screenshot. These tasks are so important. I told you that you're supposed to go and make research about the endosymbiote theory and these things of autolysis and autophagy. Yes, you're also supposed to go and try out. They're just trials, just for content check. But I want you to go and read that work again. Okay, so let me hope that's okay. Let me hope the screenshots have been taken. So I've given you the research, right? So let me give you the assignment. Make notes for today. I don't know whether you print. For me, I don't care. But I always want you to write this work on your own. Every time you write something on your own, it is easier for you to really recall it. Do you understand? That's why I want you to make notes. Yeah. Yeah, make notes for today. Okay. Let me tell you, the notes I use here are summary. So there is nothing like a summary. They're already summarized. So make those notes, read them. Read them 3 times before end of term 1. Those who don't know those who will be listening this to the future, term 1 is remaining with only 1 month to end. So you're supposed to read this work 3 times in 1 month. If you don't make it, then you're gone. Set questions and attempt them periodically. Set questions and attempt them periodically. Here I will guide you. I will guide you in holiday. And we shall post the video on YouTube. So whoever will be listening to this in the future, look for the video that really looks at how a student is supposed to read. I'll guide you how he's supposed to set questions for yourself and attempting them periodically. So you also take a screenshot for that assignment. You give it to your friends, okay? Then the research work is the other comparison, and you even have it. So you're supposed to just read and make sure you understand. Are we together? Yeah. So you will allow me sign out. So what advice can I give you for today? I decided to always have some sort of advice at the end of each and every class. So I'm Paul Tutor, a tutor of science and math at Science Scholars Academy, Uganda. You can tell me any sort of advice you would want me to talk about. So that is my what? That is my personal contact. So thank you really for attending. I sign out. Eukaryotes are done. Yeah? That means our next stop is going to be the what? The fluid mosaic model. But even when this thing, you help me one thing. Yeah? Those who are part of premium, please and please make research on the fluid mosaic model. Don't make a lot. Make sure you know something about the fluid mosaic model in the next class. I told you this one is going to be premium, yeah? Premium, you cannot find it on YouTube. If it's there, then you can't watch it. Yeah. Then I think, I don't know, we shall maybe meet in histology again or even microscopy. Yeah, but cytology, I think you appreciate. Cytology, I've covered it for you, the whole of it, and it has been free. So I think that is something good. Chemicals of life, we did something. Let's see what really God has for us for the coming days. So let me see if there is any request in the chat. Hey, I also teach physics. You didn't know? Okay, I think people don't have anything to say. Let me hear from ITEL. ITEL, yes. What do you have to say? Please unmute and speak. Yeah, those who have appreciated for the lesson, I also appreciate you all for staying for this long. Many of your friends left, but at least you have represented them so well. Let me hope all points have been explained well, so they will also understand because you've been the representatives. So hope they will not complain, and I also don't take that for granted. If you were very bad people, you would have left me here. Yes, what are you saying?
Doctor, when is the next lesson?
I will communicate in the channel. That's the role of the channel for clear communication. Is it okay?
Yes.
Okay, that is good. People also take the screenshot of this, trying to show you the whole section theory for cytology. So trust me, whatever is there, we have covered everything here. The only thing that is missing is this. So for you, you're supposed to just make research on the fluid mosaic model. Then from here, I'll come and join you here in the functions of the membrane and its parts. That's where I'll join you. That's where I'll join you. Hope we are together. Then these other things of prokaryotes, these ones, we finished all of them. Then we shall meet in histology and also have an introduction together. And then the microscopy. This one is very cheap. This one, I want to just use one class, one premium class, and it's done. Then I'll be done with term 1 work. Senior 6 work, we already have it. That means we shall be remaining with term 2 topics and term 3 topics. That is transport in humans, that is respiration, homeostasis, and then nutrition. Only 4 topics. We have a full year to cover 4 topics. A full year. Isn't really good, so good. Chemistry and math, we got done. So, I think you people allow me go to the hospital. We shall meet again, inshallah. Yeah. Okay, people, bye. Thank you for choosing SSA.